Sairam. I offer my humble pranam to Bhagwan. Good afternoon, everyone, and welcome to the final session of the two-day webinar on physics for healthcare. In the previous three sessions, we have heard scientists, researchers, entrepreneurs from different parts of the globe. It's my pleasant duty now to introduce this guest speaker of this session, Professor and Dr. Håkan Engqvist, who is one of the greatest innovators in the field of applied material science and healthcare. Dr. Håkan is a professor in material science and the director of the Armstrong Materials Academy at Uppsala University in Sweden. He received his PhD from Uppsala University in 2000 in the area of material science. He worked in the industry for 9 years developing dental materials in a venture-funded business. He spent 2 years at the innovation unit at Uppsala University and started a research group. His research interests include the synthesis, microstructure, and properties of nanobioceramic material for hard tissue repair, bone repair, skull repair, and novel methods for drug delivery. He has more than 6,600 citations, H-index of 45, and more than 50 patents to his credit. He has been involved in starting several startups and companies, and he has developed the world's toughest glass ceramic for artificial teeth and is also testing a biodegradable glue that can be used to mend fractures. His company OS Design develops implants made of ceramic materials to repair skull fractures, for example. Silox, which is another of his companies, is a spin-off from Biomedical, a Venn Excellence Center in Gothenburg, where he was formerly a researcher. Silox focuses on a variety of oral care applications, including toothpaste and gel to treat hypersensitive gums. A more recent startup, Amplicure, works to safeguard medicinal drug against misuse. Dr. Håkan has received many prizes and awards, including the prestigious Innovation Award from Uppsala University for successful knowledge transfer. He has been working on various innovations in the last decade. In one of his interviews, he says, I quote, I'm a fairly strong believer in integrating the knowledge triangle. That's how I work. My knowledge triangle comprises research, education, and innovation, but may also contain other forms of collaboration. I end quote. Despite leading many companies, Professor Håkan considers himself still a researcher. Please join me in welcoming the material innovator, Professor Dr. Håkan Engqvist. Without delaying further, let's listen to some of his great innovations. Professor Håkan, we request you to take over.
Thank you. Thank you very much for the kind introduction. I hope that you see my screen now. So I will go through, present some about the university and then some about how we work and go through some specific projects. And then in the end, I'm open for questions or so on that you might have. So I come from Uppsala, where I work at Uppsala University. And Uppsala University is, it's relatively far up north in Europe. And we are the religious center, or used to be the religious center of Sweden. And we have a dome that was relatively old. And at 1477, we got the permission from the Pope to start to teach. So the university, and this is the same as many universities in Europe, it's connected initially towards the church that the Pope gave permission to teach, and from that later on it developed into the university. to a full-fledged university. So this is an overview picture of the old part of Uppsala. It's very close to where I live. And the Ångström laboratory, where I work, is farther away in another campus area than what you see in this picture. Okay, so Uppsala University. We are quite a traditional university. It's called the Humboldt. The way it's run is called the Humboldt University. That's the same as let all flowers bloom. So there are many research groups, and we are relatively free in a way. It's a lot of tradition connected to the students. We have roughly 50,000 students in give and take some, depending on how you calculate. We are highly ranked. We are normally top 100, maybe 80 in the world, according to many of these different ranking systems that are used. Some profiles, Linné, ordering of the plants. Celsius, the thermometer comes from here. We have found quite many of the elements in the periodic table. For example, yttrium. It's a small place outside Uppsala called Ytterby in Swedish. That's where they found that element. But there are other elements that have also been categorized and found here in Uppsala. We were also the first to splitting the light, the light within the wavelength. Okay, and that, yes. And we also discovered the lymphatic system. So there are many things. We were quite prestigious, and we've done quite a few of it, especially in materials, chemistry, and medicine, I would say. Nowadays, we are very strong in what's called peace and conflict research. I would say maybe that and evolutionary biology at the moment are the two areas where we have a really, really strong position. Some Nobel laureates that we have had at the university, two of them, one of them I know that I worked close to is Kai Siegban. He got the Nobel Prize in Physics in '81 for the ESCA or XPS. I used to sit in the same corridor as him. Okay, and I then work at the Division of Applied Material Science, which I run. It's my research division. And we work at Ångström Laboratory in Uppsala, which is a relatively newly built materials or science. Nowadays also would be computer science and mathematics, but it used to be just materials. Research center in Uppsala, and it's relatively well equipped, I would say. We have access to most of the equipments that you could use for this type of research. So, as mentioned previously here in the introduction, so my personal background comes from what's called, in Europe, this is very modern, it's called the knowledge triangle, or K3, where normally researchers, and as we that are participating in this workshop conference, we are located on the crossroad between innovation and education. Okay, that's where university are, and Uppsala University is the same. Some people more towards are more towards creating how should I say research. Others are more towards educational part. But in reality, we are also then connected to, in this case, what I call innovation, but that could also be collaboration with society. And if you do this as a triangle, in this case, you can point yourself to where do you belong in this triangle as a person. Not only on creating knowledge and spreading the knowledge, but also how to collaborate and spread the knowledge out to the society. So it's a triangle. My personal view, I do not teach that much, so I'm much stronger between the creating knowledge and spreading that through innovations. So more leaning towards that side of the knowledge triangle. And why is this important? Because it creates faster societal impacts of research results. For example, in our case with healthcare, results that reach the clinic gives a higher impact. I'm 100% sure of that. Especially long term for universities, this is very important. If we work with healthcare, of course, if what we do could reach the clinic or societal use, the impact goes up. For the students, they are trained for future jobs, plus creates a good network if they are more broader in sense. And knowledge triangle normally means that you need to collaborate to reach critical mass. And it's very, very difficult to take advantage of excellent research without the integration of knowledge triangle of what you do. So this is what I'm a strong believer in. And it's basically to turn money into knowledge. That's what we do. We get tax money, we create knowledge. Next step is to create knowledge into money or societal needs. That could be, for example, create new laws. That's also a way to spread the money that we got from taxpayers back to the society. Or we could collaborate with the existing industry, or we can form new companies. For example, there are many ways of doing this. But you see what I'm after, that we get paid to do something, we create, and then I find it's also our job to help to transfer that what we have created back to the society. Okay. So more specifically, what we do in my research group, which is called Materials in Medicine, we do spare parts, regenerative medicine, mainly for hard tissue. We would like to move more into soft tissue, but mainly hard tissue. That means the dental part. CMF is the cranial, spine, joints. Yes, and we do drug delivery and some sensors, not that much. I will go through a few of these examples. And I would also say the main part, what I do as my special part is hard materials, ceramics and metals. We have been mainly working with ceramics. We are spreading into doing our own metal as well, metal alloys. In Sweden, metal and metal alloys are very, very strong. We are a metals country, and the industry is strong, and there's a very strong institute sector. So I've been thinking, I shouldn't go there, because it becomes too complicated to run projects when there is so high competition in the country. But recently we have had quite good ideas on new alloys that we are exploring, and that is both connected to our new needs, plus the new possibility with the additive manufacturing. So we are working more towards metals. I will not present that today at all, but we are doing that. So what we normally consider, what and how can ceramics help human health? How to design and prepare better ceramic-based materials. So we do structural ceramics. That means that we go for strength, optical, or stability. And normally this is a composition structure, grain size type of loop that we go through. We work with functional ceramics for bone, teeth, drug delivery, and also antibacterial. The applications are normally clinically driven, and we are always interested in better and more ceramic materials. We try to do more and better with less. So we made a small image here of what type of materials we are working with, and also describing in the let's say, the part of the periodic table that we are working with. You can see there are quite many of these of the elements, but these are the ones that we try to combine into new materials. So what do we synthesize? We do structural ceramics for load bearing. Mainly these are silicon nitride based. We spent almost 8 years on this. Quite interesting area with silicon nitride. The background is that there was one innovative company in the US who's been doing silicon nitride for spine that we have been following quite a long time. Not that much research done, but from a company perspective, they have been doing quite well, They have accumulated a lot of venture capital. So I've been following them, and then we tried also to follow their example, and we also been working with silicon nitride. We are relatively alone about this, but it's a very interesting material. It's very high mechanical strength. It could be biodegradable. It is antibacterial. It is bioactive. And so we are doing this for load-bearing applications. That means in the spine and in the joints, especially hip joints, but most likely, and I'm also interested to go for the small joints. We have developed up to the far right corner here, we have translucent strong glass ceramics. We have developed the world's strongest translucent transparent material that we are pursuing in the dental field. Okay, then nanostructured ceramics, we do, that was also mentioned in the short bio here, is we have developed a method to do small ceramic particles that we have now, is actually now being sold in tooth gel in the US. So we use it for a tooth occlusion. And then a very interesting area is to see the morphology control of calcium phosphate. How could we control the growth and structure of calcium phosphates? We have been working with that for many years. And then in the further down to the right, we have additive manufacturing of ceramics and gloss ceramics. We are trying to do, since we come from the material side, we don't from the design, we're trying to mimic different types of designs with our materials and additive manufacturing. Some applications I mentioned, spinal fusion is one, with silicon nitride. We have a lot of work done on dental, both because I worked almost 10 years in industry with developing dental materials, and then I continue at university. We do restorations, anti-hypersensitivity. We have been doing a lot on whitening. We have been doing on tooth fillings, and we spent a lot of time, actually most of the time, on dental implants. That's the titanium part that is kind of replacing the tooth root. And then functional bone grafts, I will talk more about that later today, and that is bone graft injectable materials with specific functions that could be injected directly into the bone area. And this is how it's normally done. Could be done then, it's instead of having open surgery through materials, the surgery could be done injectable instead, which saves time, cost, and pain for patients and for the hospital and for society. We have been doing a lot about this. One thing that I would like to present more about is this tissue adhesive that we are at the moment very intrigued about. And it has to do with, at the moment, there are no bone adhesives that could be used clinically. The polymers that are used in the clinic are non-resorbable, and they don't really adhere to tissue. There are a few, but they are relatively weak. The one is called Tisel. It's a fibrin type of glue that is used to glue skin and stuff like that together, but that doesn't have enough mechanical strength to glue pieces of bone. So instead, if there is, for example, in this case, an elbow fracture, that's what you see in the middle picture there. So many fragments, it's ridiculous. So they are all placed in place again with wires. So that takes a long time in surgery. And it's very hard to do this clinically. So to have an elbow fracture is complicated to fix. And this is just one example where glue would make a lot of sense. There are many things also in the foot that are complicated to fix. And fingers, there are many areas where it could be very beneficial. We can glue the patient instead of having all of these wires. So we've been working on a resorbable bone adhesive. And what we did, the first experiments were done 2013, where we glued 2 pieces of bone from the slaughterhouse. It's just animal bone that we glue together. And then just to measure the strength after 24 hours, we put buckets of water in a type of homemade mechanical tester. And we were able to do 36 kilos before it broke, before the fracture broke. So then we started to realize, actually, this is something really, really strong. And this is from a cadaver workshop where we were able to glue pieces of bone together in 2017, 4 years later. And we are now targeting to do wrist fractures, in this case, together with the screw, to glue the screw in place. So this is a regulatory question that we can talk forever about. But that's the way we're pursuing this now, and we are working really heavily on this. And I would say that if you just to imagine how many steps, so if there is a wrist fracture in a patient, these are all the steps that are needed before that patient is discharged. And then you have the payment journey. And if it doesn't heal, it goes back again. So the cost for this is around, the cost of this is around 60,000 Swedish kronor or 6,000 euros. I don't know the conversion rate to India really, but that's roughly what it would cost in Europe. And with a bone adhesive, the cost would go down to around 2,600 instead of 6,000 euros. So there is a huge saving for society and for patients, of course, if they're glued together. So why haven't that been done? And if you go and search in the literature scientifically, there are many, many people who have tried to do bone glues, many. Many come from the muscle adhesive. And some are just polymer based, but quite many, come from the muscles that attach to the boats. In the bottom of the boats, you have all these muscles that could sit on big ships. So they've been studying a lot of studies on how they are actually made, and then you try to replicate that and make it into glue. And most of these have failed in the investment regulatory human clinical trials. That's where they fail. So it's a very complicated project to run towards the clinic. It's easier to do it in the bench, I would say. And so far, no one has made it to the market with a bone glue claim. And we are working what is called the phosphoserine. And if you, for example, have a broken bone and you try to image that and mark for phosphoserine in our bone, you would see a lot of phosphoserine is already present within our own system. So we are then we are using the pure phosphoserine and added that to our material systems to get an immediate glue effect towards tissues. I normally say that what we do is an injectable high surface energy cement. That's what we do. And it seems to work. They are all degradable. So far, we have been doing rodent study, and we're going into bigger animals. So we mix it. We try 3 calcium phosphate and phosphoserine, and we're able to even measure these underwater. I don't need to go through all of this, but we made a lot of shear tests and pull-out tests on these. That's how it could look out in powder, and after hardening, that's how it normally looks. It looks more or less like a glass surface. These are just shear strength values as a function of amount of mole percent of phosphoserine. So there is an optimal between, let's say, 20 and 40% of phosphoserine in our formulation. There are many formulations that could be done like this, and if you're interested, you will also see the phosphoserine is just one part of a very big family of chemistry. The compressive strength is very, very high, and this I don't need to go through. We are testing the safety efficacy in vivo. We have now published in both ex vivo, but we also now have our first in vivo data coming out from rodents. Very complicated, I would say. very complete way of doing, because we need to develop the preclinical models doesn't exist. So we need to do them first before doing it. And I'm a material scientist, so it's quite complicated, and it's a lot of work being done on this. Very interesting project. So these are just some initial data from a femoral bone core model. And so this project is progressing. It's now, we are running it now in Uppsala. It's in Gothenburg. We have recently started to collaborate with the veterinary hospital in Uppsala, the veterinary university in Uppsala. It's in Gothenburg, and we have a lot of activities in Ireland on this with the bone adhesive. So if you have questions, you can take them after this presentation if you have any question on the adhesive. Now I move on to next area where we have spent a lot of time is the cranioplasty. And cranioplasty has a definition. It's the restoration of a defect in the cranial bone or correction of a deformity of the bone that may happen after trauma, as in a depressed fracture. Okay? And looking at old skulls, there have been in excavations and so on, there has been shown, of course, that trauma and different diseases has been common to the skull bone, and it's quite complicated to fix. So what could be done? So if you have a big defect in the skull bone, in the interim, normally you use a helmet. You need to protect the brain. Bone then could be boiled and replaced. They could be put in alcohol and then autoclaved and replaced back. So then you use your own preservation of autograft. And then people have taken the bone flap in the abdominal wall, very traditional way, complicated as you could imagine, but it could actually work, but that is normally too expensive for society to pay for. Then you can take the bone flap and put it in a sterile, may have it sterile in a -70 freezer for many months until everything within the skull bone, within the brain is fixed, and then you can replace it. And then you can just autoclave it or do ETO sterilization. Today, I would say most hospitals are using the bone flap -70 freezer model. They resorb over time. So when you replace your own bone, it normally resorbs. If the defect is too big, the old bone resorbs and goes away. Okay, so then you have a hole in your head, and this is the clinical need. So what has been used to mainly titanium and PMMA. That's the two materials that are used clinically. And the problem is the soft tissue. So what is needed is to have a method or a new material or a new design that could help to solve this problem. The number of patients are not that big. I have no idea how many it could be in India. Actually, I have an idea how many it could be in India. And compared to the total population, of course, it's rare. But for the actual patient, it's horrible, I would say. And if they have this problem that you first have had a disease, you lose your skull bone, you get an implant, and then you have a problem with that implant, you're in a bad shape. And the cost for society for each of these patients is in the order of hundreds of thousands of euro is the societal cost. So if you look at from patient suffering, that's more of an ethical, moral thing. And then it's also a real cost for society. So me and a plastic surgeon, Thomas Engström, we founded the company Ostesign. And so I've traditionally, this is how it could look like with a titanium mesh. And you see them now in place there. Let's see. Good. So what we did, we developed this one. Yes. So we developed a composite implant where we have a ceramic on top of a titanium mesh. So we use the titanium for load support. And we have with 3D printers, so it's a CAD design. We can talk a long time if you have questions about these methods. It's very, very interesting, and I would say there is a lot of innovations to be done on this computer side, not on materials as well, but mainly on the computer side, software. So we combine these two materials into one implant. And with the design, we do a mosaic design so you can have blood flow and even up the pressure from the skull bone. It doesn't need to be a high pressure. Many of these patients otherwise have a continuous headache due to high pressure from the fluids surrounding the brain. Okay. And this is how it could look like in place. What we have shown now is, I will show you a data point on the clinical outcome. We have made incisions in patients. So this is from a patient that got, he had a gun wound. In this case, I remember this, he had a gun wound. And there were something protruding. So we were able to, the surgeons open up, and then we were able to take small pieces of the implant after implantation. And we did analysis, or a lot of analysis of this, and this was after 4 years, and we can show that we have got new bone formation far away from the bone. Very, very interesting. Where you have all the components of a living bone. So, okay. So some clinical results, and these are just published data, and this was from our first 100 cranial implants. We have an infection requiring implant removal was below 2%, and the competition showed data everything from 5 in one of the cases up to even 40% in some cases. And now we have passed now over 1,000 implants sold, and the data still are in the order of this 2 to 3%. Okay. Now it moved here. Now, so you can ask, now I leave the cranioplasty and I go into bioactive materials, which is an area where I've been working a lot. And biomaterials for first generation were designed to be bioinert and not interfere with host tissue, like PMMA. Second generation were designed to form an interfacial bond with host tissue, would be the bioactive. And the third generation biomaterials are designed to enhance biological effects, to remineralize, or to be more biomimetic. And we have been spending a lot of time on bioactive and biomineralization materials. And there is an ISO standard even that one could follow. And I think there is an ASTM file standard as well on how to test and evaluate these materials. Normally, it's a soaking method. It's a very low-cost method. You do your own simulated body fluids. You soak the materials 4 weeks, 37 degrees, and then you do surface analysis, various method, and then you study what has been formed on the material. In our cases, normally we look for hydroxyapatite. The mode of action is, I would say, negative surface charge. That could be bioglasses for those who work with glasses, quite an interesting area. Calcium phosphates, and then there are some other material systems that could be used. And I encourage people to read the papers mainly from Kokubo, Japanese, very well-known researcher within the field of bioceramics. He has been doing most of these mode of action studies. I will go rapidly through these, and what we are after then is a dental material. In our case now, a dental material is that towards the host tissue form not only a micromechanical bond but a real chemical bond. So to summarize, the advantages of this is bioactive nanobiomaterials offer several advantages. It's bioactive, they have the potential to remineralize the host tissue, in this case is teeth, and the potential to hinder growth of bacteria. One last topic that I would like to talk some about is it's an area where I think maybe this is one of the 2-3 areas where I spent most of my research time is on this artificial tooth. So I mentioned the crown with the glass ceramics that we're working on, the high strength ones, high strength translucent materials. And then when a tooth is replaced, the root is replaced by a titanium screw. And reading literature, titanium has a quite good clinical outcome. But in reality, this is starting to become more and more questioned. Sometimes it's a functional replacement of the tooth, and sometimes it's aesthetics. But to form a stable interface between bone and an artificial material like, in this case, titanium, it is complicated. And on top of that, it's protruding into the oral cavity. So it's always a pressure for bacteria to enter at the gum material interface. And if that happens, I will show you a picture later on. What that means is that the bone around the titanium implant resorbs, goes away, and instead leaves like a void. And in that void, there are normally plaque formation and bacteria biofilms, and it leads to and on top of that, these patients, when you have a tooth replaced like this, there are no nerves there, so you don't feel anything. So the occurrence of what's called periimplantitis is difficult to detect. So dentists all over the world are debating what is periimplantitis, how can it be detected, the occurrence of it, and how then to treat. It's a huge opportunity research-wise, but it's also a complicated area, and I like to be I like to be that, because it's a lot of things being done, and it's a high interest area, I would say. So metal implants are skin penetrating in this case, what we've been working on, and skin penetration means a risk to future problems. So they have to form a stable unit with the surrounding tissue. That means both bone and soft tissue. There are sometimes stability problems due to poor bonding between the metal and the bone, and then you have severe long-term problems. And then there is also the lack of local blood circulation, and it's difficult to treat. And this is how it could normally look like. This is what's called a flap surgery around an implant. You can see the void between the teeth, and you see the artificial tooth on top of it, and then you see the void that has been formed due to periimplantitis. And this void could either be fixed later on, or it needs to be avoided to start from the beginning. And we have projects working on both of these. And we do the first one, maybe that could be open for collaboration in this setting. It is for surface modification. And it's to make the implants bioactive, and also, which is more complicated than antibacterial. So we do activities on materials for bone integration and regeneration. And we do that also by incorporating of active trace elements into the coating and surface modification. I'm not going to really go into details due to maybe proprietary reasons on exactly what we're doing at the moment. But we are working with negatively surface charged of titanium, and we are doing that to enhance the bone integration and reduction of infections. And we are also having projects running on when there is a void formed, how to fill this void with new material solutions. So we do also have projects on that, which is very, very interesting. Okay, so I think that was about what I planned to talk about. I think I had roughly 45 minutes to talk, and so any questions or so? I'm yes, all yours.
So I have one question.
Sure.
In your tissue adhesive material, when you're joining the two pieces of bone, there is definitely a probability of susceptibility to infection internally.
There is.
So how do you check that? Because it can be a center for extra bioactivity and extra cell growth. It's after all a substrate, new substrate that is added.
Yes, it is. You're right in that. So this is one of the, what you bring up is a good question, and it is for us complicated. So how should I? There is a new substrate, and that's that between the two pieces of bone, and we would like it to be mechanically strong for a period of time, and then magically be transformed, resorbed, and this joint, this area where they used to be should be then instead grown together and just be bone instead. So you heal the fracture. And so that's why we're now moving into bigger animals. We've been doing the small animal work, and we're moving into the bigger animals to get a more long-term perspective and bigger amounts. The complicated part to test your question in the small animals is the amount of material that is used in these small rodent bones. I don't know how, we have very good data, but I don't know how representative they are for the human size. So that's why we're now moving into sheep studies, where the size of the fractures will be bigger. So we can test that question more in detail, because if it stays too long, if our material stays too long, I think the effort that we are doing will not be successful. On the other hand, no one can really say what should be the resorption time. Should it be a week, 3 months? I suppose a year is too long. So we don't know that. We need to test.
Okay.
Yeah, no one knows the answer to that question, the real answer. That's why we need to go to big animals and test.
So just one more question.
Sure.
When you are given a different type of material in your table you mentioned, and your material with phosphoserine, the infection level, definitely what you have shown has come down. I just wanted to know, can we not, now that we have gamut of so-called nanomaterials which can to some certain extent control infection, have you tried using some of them to reduce the infection almost below?
No, not really. It's a regulatory, very complicated question to add. So we are working with well-known components, and some of the nanomaterials that are publication-wise said to be antibacterial or reduce infections. One should also know that some of these infections that arise, especially in these super big defects in the skull bone, they could have been there. Many of those are there already from the beginning. So the reason why they replace the bone is that there is an infection. So the bone is, and then just to remove that surgically, that bacteria or that infection is complicated in the operating room, and it has nothing really to do with the material. So it's already there when it's placed, and then it grows. So then comes your question, should it be something else added? So what the surgeons do, they soak it in antibiotics.
Okay.
Some of them. Some of them do that. That is nothing that the company is promoting or so on, but we do know that some do that. And then to add other components to the materials that could be antibacterial, I would say to lower the infection rate below 1%, I would say the cost and the benefit of that is low, that the cost is too high. I don't think anyone will do it. That it needs to be some other clinical application or so on, because with even bigger problems, I would say.
Okay, thank you.
Yeah.
Professor Håkan,
yes.
In this bone glue, bone glue that you mentioned, the phosphorin is a unique one, or are there any alternative to phosphorin?
Phosphoserine is a family of, there are many analogs to phosphoserine that could be used. Okay. We have been working with phosphoserine by historic logic reasons. There is a long story behind why we started with phosphoserine, but we have published, I don't have that in front of me now, but we have published papers where we describe other. So the serines there are many serines. If you search for this online, you will see there's the serines is a family of molecules. We work with the phosphoserine, but there are others like the creatine, for example, creatine. There is, if you search for it, you will find, I think that should be published now online. There is a family, and some of these are, and at the moment, some of them could be better than phosphoserine, especially referring to the previous question about the risk of the resorption contra adhesion. It could be so, okay, now we are working with phosphoserine. And it could turn out within a year or so, 2 years, I sit there and I know that, okay, phosphoserine didn't work because it resorbed too slow. We should have chosen another one of these analogs and gone with that instead. It's a money thing also. We cannot afford to run big animal studies with many of these analogs. So we have chosen phosphoserine, but it might be wrong because there are others that could be tested that have about the same effect in mechanics.
One more question. You mentioned silicon nitride based implants, dental implants,
Yeah.
but silicon nitride, if we see color wise, it will be black, right? But though it is hardest material and used in hard coatings, so is it a
Yes.
good one to use for bioimplants, aesthetic reasons?
These implants are within the body, so no one really cares if they are black or white. Within the spine, no one sees.
Dental implants I was mentioning.
No, dental implants, I think it's biologically, it's a very good idea. Clinically, I don't think they would use it by aesthetic reasons. They would go with others because they are gray in color, so they wouldn't use it.
Okay. So one more question. This antibacterial nature of this bioimplant. So what elements you are preferring for these bioimplants? Bone or anything?
It depends. It's different for different. With the silicon nitride, it's just the resorption of silicon nitride releases ammonia. And ammonia kills most of the bacteria.
Okay, thank you.
Hello, Professor Hokan. There are a few
Hi.
more questions on the Google Meet chat. So by the students. So I'll just ask one by one. The first question is, how much time does the glue take to harden?
Minutes.
Minutes.
A few minutes.
Okay. Okay. The next question is, are zirconia implants better than titanium ones?
Yes, in a way they are. The drawback is the fracture toughness, because titanium is more secure from that perspective. But I do believe that silicon nitride have biological benefits compared to titanium. That I would say, and that mainly have to do with the bioactivity and antibacterial properties of silicon nitride.
Okay. There's another question which is about nanomaterials. It says, do nanomaterials create any health problems when they are used in the body? Toxicology.
Sure. So this is a big topic. We always keep away from non-resorbable nano-sized particles, because there are too many issues to be answered with those. So, and we are not toxicology experts, so most of the things that we work with are resorbable and go away. And then the toxicity is not an issue. But I do believe if there is small particles of non-resorbable nature, it's complicated, and we don't work with that.
Okay. One more question is,
sure.
How much time your study on this biocompatibility should be done before you can arrive at at a safe time?
You mean for to go to clinic or animals?
Yes, clinic, sir, clinic.
Oh, there is in Sweden or in Europe, there is this ISO standard 13485, and normally we just follow that standard. And in the standard, they pose what type of studies that needs to be done. So there is a how should I say, communication with the notified body, and we go through, this is the project we have and what type of studies is recommended with that we perform. In some cases, it could be very little, and in other cases, it's a lot. Like with the bone glue, that's a lot. It will take, before that is in the clinic, it will take many years.
Thank you, sir. So there's one more question on my personal behalf. When you fill something in the bone, we know that as we age, the bone shrinks. So have you done any long-term studies to see, like, because the other material, will it also degenerate at the same rate, or does it create any non-uniformity or something like that?
Not really. The materials that we work, they all resorb, and they would resorb. We try now to make them resorbable in a rate that could accumulate the change in the bone, so they wouldn't be, leaving a void or stay too long. I would say calcium phosphate research have come to that level of knowledge at the moment that we know, okay, for this specific application, we should use this composition. So we create a composition that would work. And that's so no, I understand your question, but it's not really a big issue that we've seen at the moment.
Okay, sir. Since we have 3 campuses participating in this, I would just like to ask if there are any questions from Anantapur campus.
No, ma'am, no questions.
Okay. Thank you, sir. Thank you, sir, for your wonderful presentation, for taking out
Thank you very
time
much.
to be with us. We really enjoyed your presentation here.
Thank you.
Yeah. And I hope many have got inspired to take up research in this field, which is a very, very important field in medicine.
I hope so.
We also welcome you here, maybe at a time when things are better, and we'll all sit down together and listen to you.
Yeah, sure. I will come. I will talk to Sanjeevi and make arrangements. Yes.
Yes. Thank you. Thank you very much, sir.
Thank you very much. Thank you.
Sairam to one and all. It gives me immense pleasure and honor to introduce the next speaker for the day, Dr. K. Chaitanya. Dr. K. Chaitanya did his BSc and MSc Physics from Sri Sathya Sai Institute of Higher Learning, Prashanthi Nilayam, Puttaparthi, India in 2000 and 2002 respectively. Later, he was joined as Graduate Research Assistant and did his MSc Physics in Department of Physics and Astronomy, University of Kansas, Lawrence, Kansas. Then he joined as Graduate Clinical Assistant and did his MS in Medical Physics in the Department of Radiological Sciences, University of Oklahoma Health Sciences Center, Oklahoma City, Oklahoma. He later received his PhD in Biophysical Sciences and Medical Physics from the Department of Radiation Oncology, Center of Magnetic Resonance Research, University of Minnesota, Minneapolis, Minnesota. His thesis was on development of multi-parametric MRI models for prostate cancer detection based on improved correlative pathology. Further, he did his postgraduate education and training on residency therapeutic medical physics from the University of Maryland Medical Center, Baltimore, Maryland. Currently, with his vast clinical experience, device and software expertise in biophysical sciences,
is working as an assistant professor in the Department of Radiation Oncology, University of Maryland School of Medicine, Baltimore, Maryland. He is also a member of Varian Committee, Siemens Committee in the Department of Radiation Oncology, University of Maryland Medical Center. He is member of several prestigious societies such as American Association of Physicists in Medicine, International Society of Magnetic Resonance in Medicine, American Society of Radiation Oncology, Lexel Gamma Knife Society. He is also the recipient of several certificates in medicine, such as Certificate in Magnetic Resonance Guided Radiation Therapy by American Association of Physicists in Medicine, Certificate in Application of Risk Analysis Methods to Radiotherapy Quality Management by American Association of Physicists in Medicine, Certificate in Therapeutic Medical Physics by American Board of Radiology. Dr. Chaitanya has numerous publications in various reputed international journals and conference proceedings, and also has one patent to his credit. Medical imaging device rendering predictive prostate cancer visualization using quantitative multiparametric MRI models. He is also a reviewer for International Journal of Computer Assisted Radiology and Surgery from 2014. Please join in me in welcoming Dr. Chaitanya, who will be talking to us on topic therapeutic radiological physics, basic principles and current technologies. Thank you.
Dr. Chaitanya, are you able to hear us? Please unmute your Gmeet. Yeah.
Okay, very good. I think this is the most often used statement in 2021, you're on mute. So, yeah, Om Shri Sai Ram. Can you hear me now? Very good, very good. All right, so Sai Ram and welcome to all of you. It's 5:25 a.m. here in Baltimore, Maryland. My brain has just started functioning in its getting into its normal capacity right now. So, so if you give me a little bit of time while I set the stage, and then we'll get right into the talk. So I'm currently an assistant professor and staff medical physicist at the University of Maryland School of Medicine, and my primary clinical role is that I'm a medical physicist that works in radiation oncology, and but I also hold an academic rank at the University of Maryland School of Medicine. I thought this would be a good talk for all our students who are thinking about what to do next, what kind of field to get into, and when I graduated from Puttaparthi in a while ago, I had no idea about this field, and I stumbled upon this field as time went on. So let's start. So first of all, I'd like to dedicate this talk to Bhagwan. Without him, I wouldn't be where I am right now. After I finished my MSc in Puttaparthi, I went to the University of Kansas, where I wanted to do a PhD in high energy nuclear physics. I realized after some time that that wasn't exactly my cup of tea. So then I decided to change my line to medical physics, and then I got a master's in medical physics at the University of Oklahoma, where I did a lot of work in diagnostic imaging. Then I went to the University of Minnesota, where I got a PhD in medical physics. I worked in high field MRI. After that, I went to the University of Maryland, where I finished a 2-year residency in radiation oncology physics. And that's where I am right now. Okay, so this is, so the first question that can be asked is, well, what exactly is therapeutic radiologic physics? I will try to set the stage by basically my talk is in 4 parts. First, I'll talk about, well, what is the use of this field of this topic in cancer treatment. Then I'll talk about the basic physics principles, then current technologies, and then for all those who are maybe thinking about a career in this field, I'll talk about the United States perspective. All right, so how does one treat cancer? So it actually depends on the cancer type, size, and location. You can go for surgery. Surgery is often the first choice for solid tumors. Then there's chemotherapy that uses drugs to destroy cancer cells. Then there's targeted therapy that uses molecules specifically tailored to target tumor cells. For example, there's immunotherapy. Then we have nuclear medicine that uses radioactive isotopes. Then we have interventional radiology that uses image-guided, minimally invasive procedures like radiofrequency ablation. And then finally, you have radiation therapy. Now, radiation therapy, so basically you're using ionizing radiation that like photons or electrons, or you can use charged particles like protons for basically killing cancer. So how does radiation actually kill cancer? Radiation by itself can be classified as directly ionizing. So you have protons, electrons, which are charged particles, or they are indirectly ionizing, which means that if you have indirectly ionizing particles are like neutrons and photons. They don't have charge, and they cause indirect ionization. So basically what they're doing is they're liberating ionizing particles that are responsible for producing ionization, and they're also causing excitation of atoms. So if you look right here, there are 2 ways in which you can actually damage DNA. DNA is the principal target for radiation damage. And this is done by either direct action or indirect action. So what's happening in direct action is you have a photon that knocks off an electron, which then goes and hits the DNA strand, and this is called a direct action. On the other hand, you can have a indirect action where you have a secondary electron, basically the electron that is given off from the atom, and that interacts with the water molecule to produce a hydroxyl radical, which in turn produces damage to the DNA. Now, the biologic effects of this radiation, these include things like carcinogenesis, mutation, and what we're interested in is cell killing. So what happens is that the cancer cell can't grow or divide anymore when its genes are damaged, or basically when the DNA is damaged by radiation, and over time, the cell dies. All right, so what is radiation therapy? So approximately 50% of all cancer patients are subjected to radiation therapy at some point of their treatment plan. And it's largely a local therapy. And so therefore, it is highly dependent, and it's very essential that we maintain the accuracy of its, so that it's effective. And this requires a multidisciplinary team. So you have a fully trained and skillful team. It comprises of, starting with the radiation oncologists, you have radiology and imaging specialists, you have physicists like me, and you have dosimetrists. And they take an extensive treatment planning approach, with close consultation with also surgical and medical oncologists. And it's highly individualized. So which means that you have many treatments that are working in tandem. So you have a patient that is having radiation therapy, and it's also having chemotherapy on the side. And once one therapy is done, then the tumor shrinks, and then you have the surgical specialist coming in to remove the tumor. So and there's also something called as palliative treatment, where if you can actually use radiation therapy to relieve pain. Then this radiation therapy can be either external, it can be internal, or it can be systemic. External basically means that this radiation is coming from a machine that aims the radiation at the tumor. So if you look at this figure right here, this is what you're looking at in this figure is a clinical linear accelerator. So that's what it looks like right here. So you have the radiation that's coming right from the treatment head, and on these arms that you see on the side, that's the imaging part. The patient lies on a couch, which is movable, and it's a 6-degree of freedom couch. Why 6 degree of freedom? So that we can precisely align the patient to where the beam is going to fall. And this thing that you see on the patient's head is called an immobilization device to make sure that the patient does not move. So in this case, we are using a linear accelerator to treat a head and neck cancer. And that's why you have this immobilization device on the patient's head. So you have the beam coming from here, and then it falls on the patient. And it's highly localized, and so you're only treating, a tumor that is confined to, in this case, the head and neck. So that's called the external radiation therapy. On the other hand, we have internal radiation therapy. Internal means that you're basically putting a radioactive source inside the patient's body. Now, this radiation source can be either solid or it can be liquid. If it's a solid source, then it's called brachytherapy. And so these kind of procedures are done either in an operation theater, or there are specially designed rooms within the radiation clinic, within the radiation oncology clinic, where you can do these kind of procedures. If you're putting a radiation source inside a patient's body, that also means that this radiation source is going to keep on giving radiation after a while, for a while after the treatment. Then you have systemic therapy, which means that this is internal radiation therapy given with a liquid source. So the source then has to travel, to the place where the cancer is using, it basically travels in the blood to the different parts of the body where the cancer is. All right. Now, so now that I've kind of set the stage as to how radiation therapy is, and where it falls in the treatment of cancer, then I'm going to talk about the different kinds of radiation sources. Then I'll talk about the types of radiation, characteristics of this radiation, as in their dosing, and what happens when this radiation interacts with matter. Now, part of this is just basic physics, so some of you might be aware of this, but it's a good idea to get a recap. So let's first talk about radiological units. So there are a lot of things, there are a lot of units that we use, but I thought I should just talk about the few things for the purpose, few units for the purpose of this talk. So the first is electron volts. So as we all know, this is the kinetic energy acquired by an electron in passing through a potential difference of 1 volt. Now, in radiation oncology, we deal with clinical beams of magnitude MeV, or million electron volts. So that's about 10 to the power 6 eV. If you compare that with how many electron volts it takes to produce an ion pair in air, so that's about 34 electron volts, and we are dealing with 10 to the power of 6 electron volts in our clinical radiation beam. So you can imagine the amount of ionization that you are producing. Then we deal with dose. So dose is energy absorbed from this ionization radiation per unit mass, and the unit is gray. So 1 gray is 1 joule per kilogram. Now, all this is fine, but just to understand what is the magnitude of dose that we deal with in our clinic, this slide would give you an idea. So when we are treating cancer in the brain, so let's say we're doing a whole brain treatment, then we give doses of, so this is 1 treatment schema or dose regimen. So we're giving doses of the magnitude of 30 gray. So we give 3 grays every day for 10 days. If you're dealing with prostate, you're giving 45 gray. So you're giving 1.8 gray for 25 days. For breast, 20 gray in 25 days. I'm sorry, that should be 2.0. And then you have lung, 61.2 gray. But the idea being that this is what is called as fractionation, where you are giving a you're giving a certain dose for an extended period of time, and this is because of the radiobiological properties of cancer cells versus normal cells. All right, so then let's talk about radiation sources. So these are some of the sources. There are many, but I'm talking about the ones that some of them are common and some of them are actually used in our clinic. So first is cobalt-60. So cobalt-60 is a gamma ray source. It's got a half-life of 5.26 years and average energy of 1.25 MeV. And this is used in many cobalt-60 units, and I'll talk about it in my next slide. This is, for a long time, cobalt-60 units were seen throughout. They were largely in the 60s and the 70s, and even so in the 80s. They were used for external radiation therapy because you could, cobalt-60 has a very high specific activity. So which means that you can make these very small sources. And it has a short half-life, which is a disadvantage, which means that you have to replace the source frequently. So that's cobalt-60 for you. Then you have iridium-192, which has a half-life of 74.2 days. Average energy is 0.38 MeV. And this is used in the clinic as a high dose rate brachytherapy source. So it comes as a ribbon that you, so that is put into the patient and then it's retracted. So there is no source left inside the patient. That's your iridium-192. Then the remaining three, iodine-125, palladium-103, and cesium-131, these are all low dose rate brachytherapy sources. So these are used for permanent implants. So for example, iodine-125, you can use this as a permanent implant in the prostate. All right, so let's talk about cobalt-60 units. So as I mentioned earlier, cobalt-60 is a gamma ray source. It's a very high specific activity. And this is how the source looks like. So if the cobalt-60 unit, you had the source that would actually come out through here, and basically there'll be an aperture that opens when you want to the radiation, when the patient is in position. The activity is 5 to 10 kilocurie. The sources are usually replaced after about one half-life, and the source either rotates on a disc or slides in and out. But I have to confess, I have myself not seen a cobalt-60 unit, but this is how it looks like. Then you have what is called as the gamma knife. Now, when you think about gamma knife, you think about a knife, you think about surgery, and that's not wrong. You are basically conducting a surgery with radiation. So what this, what's happening in a gamma knife is that it's used for stereotactic brain radiation. You have close like 201 or 192, depending on which version of gamma knife that you have, of cobalt-60 around a patient's head, and only sources so that contribute to the radiation are unplugged. When you say unplugged, so if you remove the plug, there's going to be radiation that will strike the target inside the patient's brain. The patient wears a helmet which has collimators, and and all these sources are collimated to a single focal point within the patient. There are 3 collimator sizes available, which basically dictate what is the tumor size that you can treat. So the smallest size is 4 millimeters, goes up to 16 millimeters diameter. And the uncertainty is of the order of 0.3 millimeter. It's just fantastic. But it requires a lot of quality assurance, and this is some of the stuff that physicists like me do. We do routine quality assurance for units like these. The treatment time is like 2 to 4 hours, and as I mentioned before, the tumor size is close to like 4 millimeter, depending upon the collimator size. Okay, next we come to protons. Now, protons are charged particles, and they are directly ionizing radiation. And what you're seeing in this figure right here is, so let's look at it. So you have the relative dose on the y-axis, and you have the depth in tissue on the x-axis. You see X-rays, how the dose changes as you go within the patient. So for X-rays, there is a depth of maximum. There's something that is called as Dmax, or the depth of maximum dose, which is about here. And then you have electrons, which deposit most of their energy close to the patient's surface, or patient's body. So therefore, they are used for tumors that are much more closer to the patient's skin. Now we come to protons. Now protons, I'm sure you've all heard about the Bragg peak. The Bragg peak basically is, this is the Bragg peak. So what's happening is that the protons, the dose that they're depositing in the tissue is relatively constant until they get to a certain depth, and then they deposit all of their dose. This is very important quality, because then that means that you can use what is called a spread out Bragg peak to treat tumors at a certain depth. But that also means that if you compare the dose that is deposited from the protons versus like X-rays or electrons, you have a beam that's going into the patient, let's say a photon beam. After it goes through the tumor, it keeps on going. So you have dose deposited on the other side of the patient. So there is what is called as an exit dose for both photons and electrons. But for protons, this exit dose is not there. The entire dose is deposited at this depth, and then there's no dose deposited after that. So in that sense, protons are, they give less dose to the patient, and that makes them really good candidates for treating basically children that need radiation therapy, because we want to limit the amount of dose. All right, so the rate of energy loss, or the stopping power, this I'm basically talking about, well, what exactly is Bragg peak and how is it related to the stopping power. We talked about this. Now, how do you actually target a tumor using protons? So you use what is called as a spread out Bragg peak. So what's happening here is that this is an individual Bragg peak, but if you want to cover a tumor, then you have to spread out the energy. This is done by superposition of several proton beams of different energies. And these beams are called spread out Bragg peak beams. Anyway, this is done through something called as range straggling. Next, next we come to the bread and butter of radiation therapy treatment, and that is by using clinical linear accelerators. So as I mentioned earlier, that we are using clinical beams of the order of NeVs. But according to classical physics, the maximum K for electrons traveling at a speed of light is 255 keV. So how exactly are we able to get X-ray beams of energies of 6 to 18 MeV? Now, this is possible because we have, because of advances in radar technology in World War II, which led to the development of something called as a magnetron and klystron. They are basically RF power generation devices. So this you already know. I'm talking to a physics audience. So as far as Linacs are concerned, they belong to the S band, the microwave. And so the clinical Linacs that we use are from a company called Varian, and say they use the 3 gigahertz. But you also have something called as a CyberKnife, and that uses the X band. A little bit more about, so in order to understand what's happening in a clinical Linac, I'm going to give you a Linac tour. So we're going to trace the power path through the Linac from the wall to the patient. So what's happening here? So let me go through the figure one by one. So you have the microwave power source, then you have the circulator, you have a pulse modulator, you have an electron gun, you have the accelerating waveguide, steering coils, focusing coils, some more steering coils. There is the electron beam transport. From here, so this is basically your electron beam. Then you turn it 270 degrees, make the electron beam fall on a target. Then you get X-rays, and you collimate the X-rays. You put a flattening filter to make sure that the beam is flat enough at a certain depth, because you want to have the same beam characteristics when it falls on the patient for all the patients. That's the standard. And then you want to measure the beam characteristics, use an ion chamber, some more collimation, upper jaws, lower jaws, then when you have what is called the multi-leaf collimator. So first you have RF power generation, then what I just talked about. First thing is the modulator. The modulator is basically a big power supply. That's what it looks like in the room, and it basically is supplying power to all the devices, all the components of the Linac. And these pulses govern the operating frequency. I'm gonna skip this. This is your microwave power source, which is a klystron or a magnetron, depending upon the kind of the Linac that you have. And this is just amplifying microwaves and basically stepping it up. Then you are sending these microwaves into the accelerating waveguide. And this is basically how, there's some impedance matching going on, but I'll skip that. And this is not important. Okay, then we come to the electron gun. So this is a triode design. There's a cathode, anode, and a grid, as shown right here. So you have electrons that are being generated due to thermionic emission. And so the cathode is made out of tungsten doped with barium oxide, and you have electrons that are being released. Then they are going in this direction. There's an anode which has a donut shape, and it's basically attracting the electrons, which are then going into the accelerating waveguide. So these electrons are then injected at about 15 keV. And there's a grid to make sure that you basically controlling the electrons that are being injected into the Linac. All right, so you have electrons that have a very small, size. It's like a small pencil beam of about 3 millimeters that are going to the accelerating waveguide. Now, this is the important point. The electrons are going into this waveguide at the same time as your microwaves are coming in from the microwave power source. And what happens here? So this is an inset of what a waveguide looks like. So what's happening here in the waveguide is the electrons, as they move through the waveguide, they are basically riding. That's one way to think about it. As the electrons move through the waveguide, the parts of the waveguide, they change in their potential. So once they're in one part of the waveguide, the left side is negative and the right side is positive as they move. So they're accelerated towards in that part of the waveguide towards the end. So they're accelerated a little bit. Then when they get to the next part of the waveguide, then the polarity switches and then they're accelerated again. So by the time they reach the end of the waveguide, they have sufficient acceleration. Let's see. Okay, I'm gonna move past this. Okay, so once the electrons are sufficiently accelerated, across the waveguide, they are then going into the electron beam transport system. So there we have bending magnets which are bending the electrons 270 degrees. Not only does this, you are bending it to where you have the patient, but also any electrons that do not have the energy that you want, they're also not allowed, past a certain point. So that is what is called as an energy discrimination. The different kinds of magnets. I'm going to move past this. All right, so this is what the treatment head looks like. So you have the electron beam sufficiently accelerated, and then it falls on an X-ray target. Then there's a primary collimator, and then it falls on what is called a flattening filter. There's an ion chamber, and there's a sufficiently flattened X-ray beam, and it falls on the patient. You don't need an X-ray target when you just treating with electrons, and you have an electron applicator here, and this is the patient right there. Target in order to get X-rays is either tungsten or copper and water cooled. So that's your target right there. And why tungsten? Because it's a high melting point. And you have primary collimators. This I'm going to skip. All right, so you want to make sure that the beam that is falling on the patient has certain characteristics, but you want to measure it before. So there you use an ion chamber that measures these characteristics. And what characteristics you want to measure? Output, flatness, and symmetry. And these ion chambers are sealed and pressurized, so they're not affected by environmental variations. This is what your MU chamber looks like. Then your secondary collimator, this is how you're basically making sure that you're shaping your beam when it's falling on the patient. So you have your secondary collimator. These are basically jaws. So you have two jaws on the top and then two jaws on the bottom. So this limits the maximum field size that you can that falls on the patient. So they can go up to 40 by 40. All right, now we just talked about your secondary collimators, but if you want to closely make sure that you want to make sure that your field is able to closely conform to your tumor, you use what are called as multi-leaf collimators. So these are, you call them as tertiary collimators. They move in and out as sometimes when the beam is going through, or basically they're shaped so that they closely conform to the tumor shape. And this is how there are different companies that have different designs. So at our clinic, we use a curved leaf end, which is what this looks like. Okay, so that gives you some idea about what a typical clinical Linac looks like. And we treat with the following photon and electron beams of different energies. So we have 6 MV, 15 MV, and 18 MV. and electrons, you're going all the way from 6 to 20 MeV. Now, getting back to some of the basic principles of what's happening when your EM radiation is interacting with matter. So there are 5 things that can happen. So when your photons are interacting with matter, you have covalent scatter, photoelectric effect, Compton scatter, pair production, and you can have photonuclear reactions. Out of these, I'm interested, because of the energies that we are dealing with, I'm interested in photoelectric effect, Compton scatter, and pair production. And if you look at this figure right here, so you have the atomic number of the absorber, and this is the photon energy. What we want to know is what happens depending upon the atomic number of whatever material your photon is going through, your photon beam is going through, and what effect is going to be dominant. So you have, so if you look at the photoelectric effect on this part of the figure, as you go from this energy to this energy, you have the photoelectric effect dominant for this atomic number of the absorber. So that energy is from 10 to 30 keV. And this shows the probability of occurrence of what particular interaction. As you go further, as the photon energy increases, then you have the Compton effect that is dominant. So that's the Compton scattering. So that's from 30 keV to about 25 MeV, basically the entire gamut, this energy band. Then beyond 25 MeV, you have the pair production that is dominant. And for lower energies from 10 to 30 keV, the photoelectric effect, it's proportional to ZQ, where Z is the atomic number of the absorber. Compton scattering is proportional to Z by 8 times rho, where rho is the density, and pair production is proportional to Z squared. All right, so now we talked about the different energies. We also talked about the probability of interaction of photons and the atomic number of the absorber. Now let's look at the different characteristics of the distributions of how this dose is distributed in the patient. So let's look at this figure right here. So this is on the y-axis, you have the percentage depth dose, which shows the how much percentage of the dose is deposited at what depth in water. Why we use water? Because water closely approximates human tissue, or basically the human body. So so a lot of experiments can just be done in water. Let's look at let's look at 4 and well, let's look at a cobalt beam. So the maximum dose is deposited somewhere around here. So this means that there is what is called the skin sparing effect. So very little dose, or the maximum dose, is not deposited very close to the surface. So the skin of the patient is spared. And this is especially important because it can result in skin reactions which are not good. This skin sparing is property of clinical beams is important because as we earlier part of the century, we didn't have beams that had an energy of 6 MeV, and we were using what is called orthovoltage clinical radiation generators, and their beams would deposit a lot of dose close to the surface of the patient's body. And that will lead to a lot of skin reactions. All right, so 4 MeV, look at cobalt it deposits dose here, 4 MeV here, and as we keep going down, more and more doses deposited away from the patient's body, which is very important. And this basically shows the attenuation with depth. So why is that important? That means that if you're trying to, so you're trying to treat a patient's tumor, you can have one beam coming in from one direction, another beam coming from the reverse direction. And you get this is how your dose distribution looks like. So it means that at the center of the midline of the patient, you can get this is the kind of, you can get sort of like a constant dose distribution at the center. Now, if you want to look at electrons, so there is actually almost no skin sparing because they deposit their dose very close to your surface, the patient's surface, and which is how it should be because we are actually using it for tumors at shallow depths. So the Dmax is actually very close to the patient's surface. Now let me talk about the different techniques for external beam radiation therapy. Currently, the technique that is used most often for a lot of patients these days is called IMRT, which means intensity modulated radiation therapy. So what's happening here? So let's look at a Linac, the picture right here. This is your MLC, the multi-leaf collimator. These leaves move in and out to conform to the tumor's shape. And if you look at, this is called the beam's eye view. This is what, this is what the beam sees when it's actually going through the patient, and that's your tumor right there. And these are the MLC leaves, and they go in and out depending upon what your treatment plan is. So what happens in intensity modulated radiation therapy is that you're also changing the intensity of the photon beams as it is passing through the patient. You can use multiple fields. The Linac can deliver these beams at multiple angles, and you can also change the intensity. So you have many parameters. The more the number of parameters you have, the more conformal your treatment plan is to the tumor that you're treating. On the other hand, you can use what is called the 3D conformal radiation therapy, where you're not changing the intensity. It's just basically, you have beams at different angles. Then you have the latest technology is the technology that is used a lot these days is called VMAT, or volumetric modulated arc therapy, where the gantry is moving while the therapy is giving. So that's another parameter that is changing. The collimator is moving, or the MLCs are moving. Then the intensity of the beam is changing. So with all of these parameters that you can change, you can get a much more conformal plan. All right. Now, what happens in a typical clinic? So this is a multidisciplinary team. So this requires a lot of teamwork. It starts from the physician. When a patient comes into the clinic, you have the physician evaluates, based on the kind of imaging studies that have been done, and then decides this is the treatment regimen for the patient. Then the physician writes a prescription. Okay, this is the treatment regimen we're going to use. You're going to get this dose for these many days. Then the prescription goes to the dosimetrist. The dosimetrist designs the treatment plans and works. In all of these things that are happening, the physician and the dosimetrist works in close conjunction with the physicist, because the physicist is the technical in charge of radiation therapy in the clinic. We know the most about the interaction of radiation with matter and what the machine can do and cannot do. And we need to make sure that the machine actually delivers what we think it should deliver. So we are closely involved in every step of the treatment plan. Once the treatment plan is ready, and I'll talk about what we do a little bit later, but once the treatment plan is ready, then we have a radiation therapist that actually delivers the plan. And we have nurses also that take care of the clinical aspects as far as the patient is concerned. So this is your typical workflow. So you're starting with the consult, then we do a simulation and a CT scanner. The CT scan is of the patient is required because that's what we use to calculate the dose. That is, and this also is required to generate what are called as DRRs, digital reconstructed radiographs. The digital reconstructed radiographs are what are used to compare, let's say so you have the CT scan, we know this is what the patient, we have an idea of where the tumor is, and we have a treatment plan. Once a treatment plan is put on the machine, the patient is again imaged. And once the patient is imaged, that image is compared with DRR, which is a virtual image, and to make sure, and the patient is actually moved to make sure that their position matches with this virtual image. More about that in another slide. So once simulation is done, there's treatment planning, and there's pre-treatment verification. Basically, the plan comes to physicists. We make sure that the plan is actually deliverable. Then there is treatment delivery, and then there's follow-up. And this cycle continues because it's a multi-fraction delivery. We talked about that. So as I was talking about earlier, once the patient is actually scanned in the CT after they come into the clinic, and let's say you're treating the chest, so we assign what is called as an isocenter, or called the treatment isocenter on the patient. So that is in the patient coordinate system. The Linac has its own isocenter. What is the isocenter? That is the point about which the Linac actually moves. So you have one coordinate system, you have the patient coordinate system, you have the Linac coordinate system. Once you want to set up the patient for treatment, you have to match these. And that is matched. That's why you have the 6 degree of freedom couch right here. So you can actually accurately match the treatment center for the plan with the actual treatment center for the Linac. We have different imaging modalities that are used for simulation. You have PET, CT, MRI, and these are all used by our physicians to accurately contour where the tumor is and margins. All right, now let me talk a little bit about if you want to look at this as a career. So as a medical physicist, you have different areas of responsibility. You have clinical, research, education, as well as regulatory compliance. Your clinical responsibilities vary a lot. Basically, you wear a lot of hats. You are on constant call because you are the person who knows more about the plan, the technical aspects of it than anybody else. So therefore, that means that I'll give you one example. Let's say the patient loses weight. The patient is being treated and the patient loses weight. How do you find that out? You image the patient just before the patient is treated. You get called to the machine and the physician asks you, well, looks like the patient has lost some weight. How much do you think is the difference in the dose going to be, because we are seeing a 1 centimeter difference in the external contour? And then you go back to your training and then you try to find out, well, 1 cm, then at what point, how is the patient lying? Let's look at the patient's history. What are we treating? What energy are we using? And then you can say, well, it's going to be an overdose between 2 to 3%. Say. So all of your training comes into the question at that point. And this is what makes it really exciting. So that's your daily clinical support. Then you do quality assurance to make sure that the machines are running properly. And this is a routine affair. You do daily QA. You're looking at the characteristics of the machine daily, weekly, monthly, annually. So this is a continuous affair. Then you're responsible for getting new equipment or updating new equipment. Once the patient's plan comes over to you, you are responsible to make sure that the plan is actually deliverable. And that means that we do a very comprehensive chart check of the patient's plan. And we also use a secondary dose calculation software apart from the dose calculation software that is used to make sure that these two, that we have an independent calculation that agrees with what is in the patient's plan. You need a new site, you need to involve a physicist because we know about the shielding requirements. Then also there are state or national compliance issues. There are inspections. So, and you have people that come in and they want to market a new product. So you are also acting as a liaison between other medical professionals, manufacturers, and regulatory agencies. Then you're involved in education. I teach medical residents, technologists. We are required to give in-service to the people in the department, and there's always a requirement for getting in new therapeutic procedures. So you're always involved in those discussions. And you're in a research institution. That means there are a lot of research and development responsibilities. Because my job is primarily clinical, I'm not so much on the writing on the grant side, but I am involved in a lot of clinical projects which turn into research papers. All right, so as far as professional training is concerned, you can get into this field with an MS or PhD in medical physics, and after that you have to do a residency. Why is that required? That's because a residency has to be through a accredited program. If you do not have a residency through an accredited program, you cannot be board certified, which is an absolute requirement to practice in a lot of states. Okay, so finally, so talking about therapeutic radiological physics, it's a very exciting field. There's a challenge of applying the principles of physics to medicine. You feel that you are, you're doing something that is benefiting the patient. You can see it. You also have the satisfaction if you're developing new technology, and on the whole, at the end of the day, you feel that you're actually doing something with your hands. So I hope that this has given you at least a general idea about what the field is like and the current technologies. So that's all I have. Thank you, and I would like to thank the Dr. Gauri Shankar and the department for giving me this opportunity to share this information with all of you. Questions? All right, so it looks like there have been, I'm going to actually look into the chat window if I can.
Thank you, sir, for giving us a comprehensive idea on this field. We have questions on.
Yes, I'm actually looking at the questions right now. So first question, are there any alternative emerging technologies or trends to therapeutic radiology? So let's see, alternative emerging technologies. By alternative emerging technologies, I can talk about, in our field right now, protons are the emerging trend. And so therefore, almost every other state is trying to get a proton center. Also because a lot of people do not want to deal with this exit dose. And if you have a proton center, then you can also treat children, because we really want to limit the amount of dose that you're receiving. So I would say protons is the emerging thing. Also carbon ions. I do not know much about the subject, but that's also another emerging technology as far as radiation oncology is concerned. Next question, will the healthy cells sustain any damage during radiation therapy? Of course. But the whole idea is of fractionation, or giving this much fraction, so you are giving a certain dose expanded over, over a certain number of days, is that not only are you killing cancer cells, but you are giving the healthy cells a chance to regenerate. And that's the whole idea about fractionation. But radiation therapy has side effects. And so therefore, there's the whole list of side effects depending upon the kind of cancer that is being treated, and the kind of body part that is being treated. So of course, the healthy cells will sustain damage during radiation therapy. Okay, next question is, the lead being heavy and makes mounting them tough in collimators may not be so true, but it's also toxic in nature too. But do we have lighter and eco-friendly alternative collimator materials for gamma and X-rays? That's a good question. I am unfortunately not, I don't have any idea about other alternative collimator materials, but that is definitely a consideration, because when you're making a machine, you want, and especially something like a Linac, because it's continuously moving, to treat the patient, you want to have these collimation materials that are lightweight. What are the side effects of radiation therapy? There are a lot of, it all depends, again, like I mentioned earlier, depending upon what cancer and what body part you're treating. At the very minimal, they can be, for example, if you're treating breast cancer, there could be some reddening of the skin. But I would, that's something that, again, depends on the kind of body part. What is the success rate of radiation therapy? Well, I can see it while I check patients' plans every day, and I can see the tumor actually reduces in size. So radiation therapy obviously works. But as far as the success rate, again, depends upon, the site and, which body part you're treating.
Chaitanya Sairam, just to answer that other question by that Sai Kumar about different materials that are used for collimators. Just to probably just tell you that in our department, we are just looking at tungsten-based materials that could act as collimators. In one of the projects, we are looking at it. Let's see how it comes out. We have given some trials. It seems to be working well. So probably in the future, we could have such alternatives.
Actually, so current collimators that are used in the Linac are also made out of tungsten. So that seems to be, the industry norm as far as, Linacs are concerned. So definitely that would be something to look into.
Shivshankar Sai sir, can I ask this question?
Thank you, Chaitanya. It was wonderful listening to you, and then you have given a very, very comprehensive overview of everything, including what it is like to be a radiological physicist. Very nice of you. I enjoyed the entire this thing. I was only just absorbing that, and you've given a lot of material. Very, very well thought. Some of the questions which I also had, that tungsten material to be used, and then what is this status of these accelerator being used in India? I just wanted to know if you have any information on that.
Oh, definitely. Thank you, sir. It's actually great to hear from you. The status of linear accelerators in India is actually pretty good. There are many sites in India that are using technologies that are used in the US. For example, I know there is a
RRCat indoor.
Mm-hmm.
I don't know any other thing. In Bangalore and all, they're trying, I thought I heard, but I just want to know, because being in the field, you may know. That's what I asked.
I know that Apollo hospitals as well as Site Care, they have, I was looking at, I was doing a Google search on hospitals in India. I know that Site Care Hospital has an Electa. Electa is another Linac provider. You have Varian and then you have Electa.
Can I add one thing? So there is one at TMC, Tata Memorial hospital. So they are developing one that is ion-based accelerator to treat the tumor.
Oh, very good.
And yeah, that is in collaboration with BARC and TIFR.
Okay, very good. Thank you, ma'am.
There's actually one more thing I would like to add to Sanathan's question, is emerging technologies. Another emerging technology is using MR guided radiation therapy. And you have what is called an MR Linac. Now that is the in thing as far as radiation therapy is concerned, because think about it, you can actually image the patient. You can have an MR video that tells the location of the tumor as it is moving and then target it. So that is actually a whole new ball game as far as radiation therapy is concerned. Why this is also important, because if you look at a CT image versus an MR image has more soft tissue density. You can see more things. For example, if you look at a prostate, you cannot see anything in a CT. You cannot see its contours at all. On an MR, you can see the contours, you can target it better. So that MR Linac, there are 4 consortiums as far as I know. You have MD Anderson, then you have, I believe, in Belgium, you have Australia, and also Canada. So that I would say is the next emerging technologies.
Thank you.
Saram sir, thank you so much for such a wonderful presentation. I just have one basic doubt. You had discussed about the fractional dosage so that the healthy cells, we give a chance for them to sustain any damage. But don't you think such an approach also increases the risk of metastasis? Because you kind of damage the cancer cells, not immediately, but you treat them over a period of time. But there is a possibility that the cancer cells are broken down into smaller pieces, enter the bloodstream and get attached to some other site in the body. So metastasis becomes a big problem there. So any approach to combat metastasis? I know it's a very fine line because you don't want to damage the healthy cells. At the same time, you don't want metastasis to happen. So any approach to that?
That's a very good question. So first of all, external beam therapy is a localized therapy. So you're only targeting what you can see, which means that so you target a tumor, then you do constant imaging, and then you find out, okay, well, the tumor is shrunk. Okay, very good. If there's a tumor that has gone to another part of the body, then we'll try to look at that, try to address that point. So as far as this risk of metastasis is concerned, that is something that you cannot really control. At the end of the day, unless you are you have to target the tumor. That is, as a physician, that is your first that's your first idea, that okay, let me take care of the tumor first, because I have heard this multiple times in many of these chartrons that we have when we discuss patient plans, is what is more important right now? You have a tumor that will probably kill the patient, or do you want to you have to address that first before you try to look at the different mets that are generated. That's always a risk. And that comes with the fact that you're dealing with radiation, and radiation kills. But as far as tumor control is concerned, what are your options? Depending upon the kind of tumor, the type, the size, can you take it out surgically? Maybe not, because of the patient may have other complications. So that's why when I said initially that radiation therapy is part of multiple things that you can do in order to target cancer or look at cancer. So the risk is always there.
So one last question. You are first of all, it was nice taking us back to the undergraduate days to see all the nuclear physics concepts and revising them, all the accelerator physics. And on the other part of the question that I want to ask is, you showcased so many of these technologies, but as an end user, we have so many types of cancers coming up. So in a very nutshell, or in a bird's eye view, if you can tell which is the gamma knife or other radiation therapy, can we target and tell us for breast cancer, I use this. Can you tell us like that, because you are the expert in this field?
Okay, so so basically what you're asking is if there is a certain modality that is suited for a certain type of cancer, right? That I can say that, okay, when you have this kind of cancer, use this modality.
Yes, sir.
Okay, so let's talk about the different ways that, for example, in the clinic that you can treat, let's say, prostate cancer. So one is, what is done in the clinic is you can treat prostate cancer by external beam radiation therapy, or you can use what is called as low dose rate brachytherapy. In low dose rate brachytherapy, what is going on is the patient is in the operation theater. You're using an ultrasound to visualize the prostate. Then you are implanting permanent iodine 25 seeds inside the patient. Why are those seeds implanted? That depends on the kind of cancer that this person has. Depends upon, when I say kind of cancer, it depends upon the staging. So depending upon the Gleason score for this prostate cancer, you can use this modality. On the other hand, so in this case, the cancer could be localized or confined to a certain part of the prostate, or you want to target the whole prostate. Then you use external beam radiation therapy because you're able to target the whole prostate. If you want to target part of the prostate, you can do low dose radiation. Then, if let's say you're talking about the brain, you can choose the gamma knife. So I talked about this earlier. I would say that the gamma knife is the modality that you can use for targeting tumors that are really small. You can target tumors as small as 4 millimeters. So I would say that would be so gamma knife is the modality to target tumors that are this small. So to answer your question, there are actually, depending upon the tumor size, I would say brain tumors, gamma knife, definitely. If things that don't have gamma knife, there's also Linac alternative. We are limited on the Linac by the size of the collimation that you can do. On the gamma knife, you have a very small collimator, like a 4 millimeter collimator. Just open the cap and the beam goes in and you can treat the patient. But on the Linac, you're actually dealing with not with a radioactive source, but a clinically generated beam. So that beam has its own characteristics. And you have, like we mentioned earlier, a tungsten collimator. So if you want to treat on a Linac, you can actually use what is called as a cone. A cone is like a collimator with just a slit, very small slit. But you need a special cone device that you can attach on the Linac. So to be honest with you, there are so many different ways in which you can treat the cancer. It depends upon, highly dependent upon the site as well as the type of cancer, how fast is it advancing, and things like that.
Thank you, sir. Thank you very much.
No problem.
Thank you, sir. Thank you very much for taking us to giving the knowledge about radiation oncology and the workflow in radiation oncology.
Sure. And please, you can always reach out to Dr. Gauri Shankar if anybody wants any more information about, how to get into this field. I'll be more than happy to help out.
Thank you for taking out for your early morning time for us.
Yes, I'm all awake right now. So.
Thank you.
Right on.
Om Shri Sai Ram. I once again thank the speakers of the afternoon session. Over the last 2 days, we have been listening to eminent people from across the globe who have talked about various aspects of physics that are being used in the healthcare industry. We have had talks from eminent scientists, academicians, industry heads, and entrepreneurs who have taken us through this journey and showed us how physics plays a very prominent and definitely a very important and pertinent role, I would say, in the field of healthcare. But I would also say that what we have heard, what we have seen through these talks is probably only a glimpse of what the physics can actually offer in this field and the connection that physics has with healthcare in the global scenario. And so I'm sure there's a lot more to learn, a lot more to understand from all this. And with the advancement of the science and technology, there are more and more newer devices, compact handheld solutions, systems, integrations, and of course the artificial intelligence that's coming in to play a crucial role in all this. So because of all this, there is a larger aim to make the healthcare itself more affordable and more better with the advancement of this science and technology. And we all know that the governments across the world, all the governments are pumping in a lot of money in this particular process. So to know more on this aspect and how healthcare technology is changing, what opportunities are there for physicists as such in terms of funding, in terms of youngsters who want to pursue PhD, and how governments are supporting this endeavor, and probably how governments are even supporting the entrepreneurs and startups. We now have a panel discussion on the topic changing landscape of the healthcare technology. We are grateful to the members who have agreed to be in this panel. We have 5 panel members who will be having discussions on these issues. I now request Kumari Shushma to introduce the panelists, after which I request Dr. Anand Krishnan to steer the discussions and take over the proceedings of this panel discussion. Thank you.
The next panel member is Dr. Saisham Narayan, currently working as Technical Manager at Biotechnology Industry Research Assistance Council, Calicut. Dr. Saisham completed his BSc Biosciences from the Sri Sati Sai Institute of Higher Learning. He has worked extensively on production, purification, and biochemical and biophysical characterization of recombinant Mycobacterium tuberculosis enzymes, which are potential drug targets against TB. His current research is in the area of telomere biology using techniques involving genetics, molecular biology, protein biochemistry, and structural biology to answer some of the basic questions in his field. We also have Dr. Vandana Sharma, Associate Professor, IIT Hyderabad. Madam has obtained a PhD in Experimental Atomic and Molecular Physics from the Physical Research Laboratory, Ahmedabad. She was a postdoctoral fellow of the Max Planck Institute, Germany, and served as a guest scientist, University of Duisburg-Essen, Germany. She has been honored with various awards and distinctions: the Sri Ramakrishna Paramahamsa Research Grant Award, Max Planck Society Fellowship, Sheldon Dart's Best Scientist Award in International Conference on Photonic, Electronic, and Atomic Collisions, to name a few. Her major area of interest are experimental, atomic, and molecular physics, and design and development of ultra-short and femtosecond laser optic devices. She is recognized for her contribution in designing charged particle imaging spectrometers and medical equipment development. Finally, we have Mr. Satyadeep Vishwanathan, lead member of technical staff, Big Tech Labs Private Limited, Bengaluru. Mr. Satyadeep completed his BSc Physics and MSc Physics Electronics from the Sri Sati Sai Institute of Higher Learning. He is an electronics design engineer with over 14 years of experience in biomedical research and development. He is experienced in designing analog circuits and optical systems for portable medical diagnostic equipment. Mr. Satyadeep was a part of the research and R&D team at Big Tech that realized state-of-the-art devices like TrueLab multi-PCR platform, TruePep Auto, and Beagle Z. I now request the moderator of the panel to take over and open the discussion. Thank you.
Sai Ram, good evening, everyone. Big and warm hello from Andhra Pradesh MedTech Zone. I sincerely thank the Department of Physics, Sri Sathya Sai Institute of Higher Learning, for conducting this 2 days international webinar on physics for healthcare, and I'm pleased to be a part of this event, even if I was just here for the last leg of it, but I'm privileged. Thanks to Dr. Gauri Shankar and Sister Sushma for introducing me, and thank you for all the panel members and all the audience. So before we go into the discussion, I'll just talk quickly about the timing of the panel. So I'll just spend a few minutes on the theme of the panel and theme of the panel, and then questions. Directly to go to the questions. I trigger questions that came in my mind, and there's an interesting part here because as an engineer, my brain is black and white, and as some of biomedical or medics, their brain is more color spectrum. So so they have a lot of clinical problems. Among engineers, engineers can always give solutions. So when they both go to a table, they bring out innovation. So I would start with the question, and so the subject matter expert can start the thoughts, and slowly we can go around the panel. And as the last segment, we'll get the questions from the audience and conclusion for that. Okay. So it's really interesting time of year where we see rapid changes in the medical health technology. Right? If you really think about yesterday, it's really more like a sick care than a health care. So it's more reactive, and the data we get is quite intermittent. We wait for the occasional blood pressure check, vital check, EKG, and that makes our health technology very discontinuous. And we wait for doctors, and after a long wait, the doctors gives a flashback, need to understand the flashback of our history and give a paper-based prescription. So, but this is changing. We know we are in the transition. If you go to the remote part of the villages, the doctors still do that. And as when you go towards city, and you see that things are changing. There is a new way of connected health that's happening. Digital health, genomics, mobile makes us rethink the healthcare. We can use technology to change from reimbursement-based medicine, which is like a sick care, and now we're going to go as an evidence-based medicine, where even before you fell ill, you know that you may may fall sick in the next 2 days. So we have technologies going away. So the doctors nowadays, we're on the verge where the doctors can stop becoming God, and you became the CEO of your own health. The scenario is rapidly changing. So this panel is really what I say is, as the healthcare is changing very fast, I feel sitting in this panel is like me taking a break on a highway in a service station and think what's happening and how speed we are going. So the theme of the panel is therefore changing landscape of healthcare technology. So the goals I see is we really like to simulate the thoughts and discussion whirling around this theme, and we influence some facts and opinions and plans on this theme. So since this is an educational panel, it's not a public panel, so we would like to exchange conceptual knowledge, clarification of certain theories, because one I could see is a so varied group here. We have experts in analog design, and we have experts in biochemistry, and we have microscopy, and we have lasers. So it's as one became an expert, the other becomes less ignorant. So we got to adjust each other, and then let's clarify the theories we talk about, and let's use as fundamental as the language could be, because we also have students around. So I really like to have to focus our topic. I'll really have encourage discussions on latest trends of instrumentations, and I could see say Sri Satyajit Vishwanathan. So you are into or you're more into the instrumentation side, and also mathematical modeling, imaging, simulations, and how the point of care devices is evolving towards healthcare. And I will also like to touch upon the artificial intelligence, the future, and also how all these rapidly changing medical technologies are also going to impact the regulatory and policies. So the policy making bodies and regulatory bodies need to change at the same pace. So let's have some discussion on that too. And lastly, to make these all happen, we need our students, and we need the future students. They need to know where the opportunities are. So you being experts in your field, shine some light on it. So the researchers, the budding engineers can understand where they can focus and where they can channelize their instructions. Okay. So I would say, let me put my first question to the panel. So in your own domain, how it could be your domain or your own perception. I'd like to go around the panel, see how you think the healthcare has changed. So this could be probably 3 minutes around each of us. I would like to start with Dr. Vandana Sharma, if that's okay.
Okay, so the place where I am from is, we are actually located in it's not really a village and not really a big city. So if we go and see the doctors, so generally it takes almost 1 to 2 hours to really visit the doctor. But in the time, in that 1 and 2 hours, it's not like that we are not entertained at all. So there are various things, various steps which has been already put up by the hospitals, wherein they start taking from weighing yourself to the BP monitoring, and everything is being checked up, and then what doctor sees is almost everything, every detail which is already done by the his or her assistant. So I mean, if I compare it from the beginning, so from my childhood, it's really actually changed tremendously. Where most of the things were done by the experts only. Now because most of it now has become automated. Like the BP machine has become, most of it is automated. Even if you want to see that, nowadays, as soon it will be like sugar machine is going to be automated. It is not you don't have to prick it. So it is. We as a researcher, we are also working on that part so that we can make it non-invasive. So things are becoming. Starting from invasive to non-invasive, and making from manual or mechanical to automation. So I mean, this has really filled the gap between a patient and the doctor. So they, I mean, doctor can interact more. The doctor can interact more with the patient. So he can look at he doesn't have to spend more time on really finding out what problem the patient has, because most of it is becoming automated. So with that, I mean, more and everything is on the doctor's screen. So it's like every detail of the patient is on the screen, and he and she can actually see what is there in front of what is the person, the patient who is sitting. So he knows all the history. So then he can spend more qualitative time with the patient, and he can explain what kind of a problem a patient has. So this automation, I mean, the way it is changing now is it's increasing the interaction time between the patient and the doctor. And that way it's becoming better and better. I mean, eventually, if I compare from maybe last 20 years or maybe last 30 years span, so it is really changing tremendously, and slope is really very, very steep. So I mean, this is what I would like to say. Yeah, this is how it is taken.
We are seeing a lot of automation, and there is an increase in more than the travel time. There's a lot of interaction time
yes,
possibly happening. Yes, sir. So can I ask Dr. Sanadana Kungulu, please? Sorry, I think you're muted, Doctor.
That's correct. Thank you. Thank you, Anand Krishnan. Thanks to Gauri sir and for the invitation, and also to the Department of Physics, SSSIHL. At the outset, means we are at a very interesting juncture of the evolution, the 21st century, where things are driven mainly by data, and also we are looking at sustainability of what we are doing. And when we look from that particular perspective, even the medical device market is changing a lot. The reason why means the life expectancy maybe 300 years ago probably is 40 years. Now you see 80 years or something. So the disease that we didn't know before is kind of cropping in now, because you live longer, you tend to have much more complications with the human body. So the technology, the good point is our life expectancy is increasing, but at the same time, technology as well is evolving at the same pace, maybe a bit more faster. So with the advent of new science like OCT or photoacoustics, there are lots of new things going on, the fundamental science, and artificial intelligence, machine learning, which can efficiently handle the data. And with the policies across government agencies and the states to have sustainability and the inclusion within, I see there is a big change that is happening in the landscape of healthcare industry. So set this from researcher perspective or from university perspective, it's not that simple for us to sit and say, okay, means in 40s they worked on radiation and basic optics. Let me teach our students exactly this. No. What I really feel is that with changing landscape, especially for universities or the state body or decision maker, policy maker, they are doing it already. We have to sit at the forefront, keep monitoring the technology day in and day out, and look at the current trends in the market. Just for me to give an example, I am from the biophotonics, like light-based healthcare sector, where we think what's the size of the imaging technology. It's around $100+ billion. You will think most of it is MRI or CT scan. The truth is not that. 2/3 of it, around 70 billion, corresponds to imaging, that is photonics. And in that, you will be surprised to see 1/3 of that is endoscope market. It's like 30 billion or 40 billion. So means when we make the syllabus for our students, or when we make our goals to reach this sustainability or afford the data that we are going to get, means we should be very strategic here, look at the current trends, try to incorporate into the student syllabus and so on, so that we are creating real future leaders at our universities as well as in our state or country, to create an impact as we look forward to our glorious future.
Thank you, Doctor. Thank you very much. And Dr. Saisham Narayana, can we have your input?
Thank you. I hope I'm audible.
Yes, sir.
Sairam, and thank you, Dr. Gauri Shankar sir, and all the faculty members for inviting me to represent BIRAC actually in this panel. So coming to the question, how healthcare has changed. So I would say it has really changed in the past decade, definitely. And I would say it in a very positive way that, now doctors, they treat their patients mostly, quite objectively. There's very less of empirical treatment that's happening. So meaning that they would need support from instruments that would give them the data which they can rely on. And more actually, doctors are actually relying on this kind of data driven treatment, and they don't need to even see the patient in many cases. So like, now we are moving towards telemedicine and digital health and these kind of things. So definitely things have changed. And I think from the policy side as well, now with the Ayushman Bharat, now I think, and more more attractive policies that have come from the government side, and with the change in the medical devices and regulations and rules. So there is a lot more interest in the country, especially from Indian scenario, for developing more medical devices indigenously and to address. And especially I think I should make a mention, like, for a country like India or in the Indian subcontinent, I think having affordable and accessible solutions are the most crucial. And we still do not have that. This is what we understand in the rural India. So that's where I think, that's where the change has to reach, the ripples have to reach even there. I think things have changed and changed for good in the cities, but I think this needs to actually get to the periphery as well. Yeah, I'll stop.
When I do. Sorry, you have anything to add, sir? I interrupted you.
No, thank you. Yeah. I leave.
No, I agree with your point. My wife, she works for a medic, and so he is a rehabilitation consultant, spinal injury consultant as well. So I do know that 5 years ago, almost 5 years, they've been talking about getting everything into telemedicine. And past 4 years, the movement has been slow ever since COVID came, and suddenly they had time to do everything. So now, so I would say at least 60% of his consultations happening virtually, and and the thing is, there are so much scope to improve the telemedicine tools, but it's just it's in progress. Within 3 months, they managed to get their alpha of the telemedicine tool, and now they get working on this beta tool. I agree. There's a lot of interest from the nation and from the government, and also so much direction towards telemedicine. I would like to ask Sri Satyadeep ji, please.
Yes, Sairam, and very good evening to all. So basically, I would like to speak in terms of what we have been focusing on at my workplace. So basically, we saw the need for gold standard diagnostics at near care locations. So near care meaning to say near the patient, that is what is also called as point of care, or within a short distance of travel for transport of sample or for the patient to travel. So sample especially for diagnosis of infectious diseases. So we basically, you see this basic labs, the primary health centers, for example, they have been equipped with mainly basic methods like smear microscopy, CBC, complete blood counting, culture, etc. So we saw the need for molecular diagnostics at primary health care centers. So basically, this would eliminate batch processing, and with rapid systems, we could get the results out soon, so that the treatment for patient can begin early, and the patient can be on the road to recovery quickly, and that in turn prevents the spread of the disease. And the main challenge has been to come up with a device, which can operate in resource limited settings, meaning where you don't have continuous power or infrastructure for preparing reagents, etc. And we require a very trained manpower, especially for molecular diagnostics. So we came up with a platform which requires less hands-on training. So the sphere microscopy lab technicians can use these devices. So, and in terms of engineering, the challenge has been miniaturizing the circuitry, the analog circuitry, power management. These are actually battery operated, and also for optics design. So the optical components, especially, they tend to be it's difficult to procure them. They tend to be expensive, and most of these are imported. So that is the basic thing. So in 2018, there has been in Andhra Pradesh state, it was implemented for a TB diagnostics. So in collaboration with AMTZ. And we can do live monitoring of the data, the patient's data. So that's another feature. So that has been the kind of our focus.
Excellent. Just you should forgive my ignorance. This molecular diagnostics, when you say molecular diagnostics, you said it eliminates batch processing, and the results will be appearing soon, and the road to recovery is much quicker. But what happens exactly in molecular diagnostics? I know quite a few biomedical people around. They may understand it, but I'm an engineer, so if you can, especially production engineers.
Yeah, generally you see in the centralized labs, they have these benchtop machines where they process 96. They have these 96 well devices, basically these RT-PCR platforms. So as far as I understand, that takes time. They have to batch the samples, wait for them, because if you run the samples individually, it's not economical.
Very true.
So our machines, they can process individual samples. It's a low throughput system. It can process individual samples. So as the patient sample comes in, and the sample to result is 1 hour basically. So that's how it helps.
And when you said the challenges are in terms of miniaturization, and you also mentioned optics, is it you mean the having in the need for bulky optics makes the miniaturization less possible?
Optics,
Okay.
bulky.
Okay, and I guess the power management, battery life, that's another aspect, isn't it? Okay, wonderful. So I do have some inputs. You have anything else to add, Satyadeep ji?
No, that's it for us.
Okay, no problem. Yeah, so so from my side, I also have I see there is a lot of shift in the way the innovators even think. I'm sure innovation doesn't happen when you're busy with something. You're between activities, you get this gap, and that time you have this spark of thought, and that is the time you think of social problem and how I can contribute, or what can we do about it. So if I give you statistics, we have about 18 within me we have about 18 incubators, within Medi Valley, and I would say at least 8 of them. That's about 55% of them are working in this COVID related. So that's not only that, most of them are into medical devices also. So it's not like COVID is all about virus, and then I don't care. That attitude has gone. Most of them are engineers. All those 50% incubators I'm talking about, startups they're talking about, and so they're all engineers. And all their projects are more about point of care, multi-para monitors, ventilators, PAPR systems, and smart IoT masks, and and endoscopy, video laryngoscope, those sort of projects. I see this is it's somehow fortunate people who escaped COVID. They managed to direct their innovation to be problem solvers. So I see there's a shift in that aspect. Okay, in the way the innovators think, they're all concerned towards, they're all directing their thing towards the healthcare. Okay, and I do have next question, which is a totally different one. When I'm not sure how it works in India, but I lived in UK for a long time. So first year of your car insurance will be really high because you're a new driver. Okay, and then you show the government that you are a good driver, then they reduce the car insurance premium by half. Okay, one offense you do, it again shoots up. Okay, and again they started bringing this tracker system in your car, where based on how you maintain your speed, someone somewhere once in a while they monitor your speed and then report to the insurance company that he is a well-behaving driver, or the one who might disobey law occasionally at night times for some reason. And so that sort of tracking, as when I heard that, I felt, oh, this is very good, but something shocking also I feel. What happens if those sort of point of care, not point of care, those sort of tracking devices get into human insurance, health insurance of, instead of car insurance, think of human health insurance. If we have devices which monitor us, what time we sleep, how we look after ourselves, and then that gives premium for our policies. So I do know there are a few companies working in this technology, but two things I like to know, just from the commercial point of view, how well this is going to be, and also from technical perspective, what are the technologies that will make this happen? Okay, and from what I heard from Dr. Sai Shyam-Narayana and Dr. Vandana Sharma is like non-invasive instruments are up and running. If these instruments start talking to each other, then we are monitored for how we look after ourselves also. So first, let me ask what technologies would drive from your perspectives. So can I ask Dr. Sai Shyam-Narayana, what is your perspective on this?
Very interesting thought, actually. And then I think you have already hit the non-communicable diseases plus connected health. Somewhere, I think that's where I think we have reached by this discussion. So I think, this would become very interesting, actually, once we start discussing this. So your question was, can you just repeat the question, the final question that you had?
Question is just I do know, because this, if this has to happen, where your connected health, right, if this comes a reality, and this is going full scale, and yeah, what are the technologies, or what are the scientifics contribute to it? Yeah.
Right. So yeah. So I got your question. So yeah, we are. So we already have some of these things, which I think this thought process can already be implemented. So we have this non-contact or contact connected devices, which actually works like we have lab in a box kind of systems, which can actually be work to these kind of modalities, where I think it can track you inside your home, maybe, or I mean, as you go, you don't need to probably wear it. Maybe you can have it under your bed, a non-contact thing that probably measures your how long you sleep and how well have you been behaving and things like that. So this is definitely going to improve the life. When if somebody is actually he cares more for his insurance payment than his health, definitely this is going to be actually a good feedback for him. And yeah, this is definitely doable. It's a great idea. I mean, I'm sure, as you said, there are few who are already thinking about it, but I don't think it has reached this concept per se hasn't reached our healthcare insurance sector. This is my understanding. You can correct me if I'm wrong.
No, I just straight away this analogy came. It's like having a police in the home is your family, and having a police in your pocket is becoming like that.
Some of that.
Yeah, I'm thinking what happens in European countries is basically the police, the law is so strong, the punishment is so strong. Until you're good, everything is good. The moment you start doing bad, the punishment is so strong. Then we behave ourselves. So, so here, I think this is like this. Monitoring is having a police in the pocket. Vandana ma'am, anything?
I would like to add something to it. I mean, it's good to be monitored time to time, but the thing is, the way as long as generally it's I'm just trying to explain the things from the physics point of view. So as long as we are in the visible range, or let's say in NIR range, we are safe. The moment we are going towards really the bigger waves or the ultra short, the bigger waves in the sense the bigger wavelength and the very, very short wavelength, then we are actually increasing the risk by monitoring our health. Remember that. I mean, the radiation, we have to be in a controlled zone where we are being monitored with a safe radiation. Okay, so I think I have already communicated this. So I mean, it's always so when we are making anything non-invasive, we have to be careful that are we really following that protocol, the radiation laws and all. Definitely, everybody, all of us are trying to do that. But let's say if we even if we keep on being exposed to certain radiation, which might not be harmful in a shorter time duration, but it might be harmful for if let's say you are being exposed almost daily. True. Yeah, so then we have to be careful for that also. So I mean, yeah, this is technology wise it is good, but so should we we should be asking ourselves that should we be monitored daily, or it should be like maybe monthly basis or quarterly basis.
True. I mean, it just tells me is non-invasive really non-invasive?
Yeah.
And, secondly, that also gives us a lot of R&D scope within the spectrum of between the visible range. I would say visible range to near infrared range. How all your point of care devices or multi-para monitors, non-invasive monitors, glucometers, everything that can work within the zone.
Yes.
That's good. Okay. Thank you. And Satyadeep ji, you have anything to add here? Oh, sorry, one minute. Oh, no problem. And Sanatana ji, you?
Sure. It's a very good thought and good trend that is going on, and the technology is in line with this trend that is seen. That is, if you want to monitor people, you need, as Vandana ma'am is pointing out, non-invasive tools. And there is a big trend of optics. So it's not before. Now the optical technology, or means which the visible and near infrared region, are getting matured more and more. And they are moving from lab devices that are size of the room to wearable that you can wear in your hand. Like for example, the wearable sports watch that you have, which monitors your pulse as well as blood oxygenation. So these things, of course, going to help or solve the technological perspective of it, in the sense like they are getting mature day by day, and they are in a position to provide so many vitals for the insurance company to make any possible monitoring or tracking. But there is also need, and which there is a need for the maturity of how the data will be maintained. Of course, what happening with my personal life, all my healthcare shouldn't be story for everyone to see. Means it should be much more secure, and also we should see how insurance work. The insurance work where everyone chips in some money and it's equally distributed to all. So if you are going to punish already a person, means not because of daily habits, but because you are seeing an irregular pattern in his health, though he follows everything, that means the idea of insurance is lost, in the sense like you are going to ask him more money, which the insurance is more like you don't know certain things where others can help for a common uncertainty that is same. So I think there is a small minor circle that has to be matched and has to be built properly.
Very good. I we did come across, me and Dr. Jitendra Sharma, we did come across an inquiry from an Australian company, who are into, knee prosthetics. Post surgery, there is just on your knees you wear a belt, which constantly monitors. I know in my own house, my mom, she had a surgery, and then what happened is she was constantly complaining, what did these doctors do? I'm still having the pain, I'm not relieved. But whenever we go for consultation, the doctor asks one question back. After a post-surgery, I asked you to do certain exercises. Did you do? The answer is no. So, but this gadget will keep in track and keep vibrating when you not vibrating. It just keeps popping. You'll have notification on your mobile that you haven't done your job, you haven't done your dharma. So, those sort of devices definitely handy. And
absolutely, this is interesting as well. Just to add to that, my current area of research is exactly in those directions, in the sense like Antonio presented yesterday, where we are building actually non-invasive tools to do post-surgery monitoring, but also for physiological monitoring or healthcare monitoring or sports person. So it's like, exactly. So non-radiative, non-invasive means the technology is being built, and how it's going to be innovated and used for public healthcare, that has to be, it's not exactly our field of domain, but it has to be looked with the same meticulous or circular level, how it should be used from the policymakers' perspective.
True. Just before I move away from this topic, it's still not connected to insurance, but the point I want to do is how how these can work in and how basically monitoring someone remotely. The company is monitoring here remotely, the insurance company. But there is a interesting project I heard from NASA. It's called BioWest. Okay, it happened in about 2010. Where the astronauts, they're sleeping in space, right? So if you look at their life, they go around the sun 16 times. They orbit 16 times. So basically 16 set of sunsets, 16 sets of sunrises. That's a pretty bad jet lag, right? For someone to have. So, so they have this vest, which is like a BioVest, which picks up the ECG, the heart rate variability, and then understand the sleeping patterns. So this technology existed in NASA about 2011. So interestingly, it's now coming to us here as well. Okay, okay. The last, no, not the last. I mean, probably one more topic I would say. We'll go a bit technical in this. Experts who are all into mathematical modeling, algorithms, simulations, especially in imaging, and how you do optimization. So how do you see the digital image processing? What are the challenges you have? This is something always on demand, and starting from the MRI time to still now, any progress you make in imaging, it always get observed, get into commercial side. So how do you see this algorithm development and soft simulations? How is this field emerging? How are they now directed towards healthcare? Is anyone has any inputs on this?
Sorry, I don't work in this area, but we are trying to get help from image processing, and there are. I mean, it's very. I don't have any knowledge about the image processing, but the way we are taking some help, we have obtained some images, and we need some background to be subtracted. And I mean, you won't believe the way image processing works is like it really removes most of the background. It almost removed most of the background, and now I can actually see what is under my skin. So I'm able to see that. So that's, I mean, doesn't matter if you have hair on top and you are getting the images of the hair. So image processing has it has been evolved so much, which I don't know what tools, what algorithm they are using, but the way they are using, so they are able to subtract all out, and then it's like you have peeled off the skin and then you have seen inside the skin, and that is just with one image what we obtain.
And they are non-invasive also.
They are all non-invasive. Yes.
Wonderful. Yeah. So who else work in this field, if I may ask? Is Dr. Dr. Saisham Narayana, are you in image processing? I know you're into microscopy and molecular biology. Okay.
I can add. Yeah, exactly. I can add a bit here. Means because when we say image processing, means I see two things. One is the processing power. Another thing is storage power. So there is a lot of evolution. So in general, certain fields or domains, it means like they become very popular or they thrive when they can take advantage of other domains where the technology growth is happening. For example, in computers, there is processing based on GPUs and parallel processing on multiple threads are getting very famous and available. This is what is driving the field. And to say CT scans and MRIs, these are the things, in my opinion, needs less processing power because they are simple, straightforward back projections. It's not that straightforward, but compared to what we do in diffuse optics. So when you say the X-ray through the body, it's transparent. You can make the imaging, like by doing a simple back projection. In diffuse optics, like our field, where we use visible light, we are not transparent beings. Means we want to know every photon, the visible light or NIR light sent through the body, which path it took, and accurately make a clear-cut algorithm based on a lot of science and physics of photon propagation in the medium to make a reconstruction. For example, OCT needs some computational power. On the other hand, there is an emerging technology called photoacoustics, which uses the contrast of light and the resolution of ultrasound. That needs a lot more power. And there is emerging technology that we work acoustoptic, where with the processing power is a lot more needed even. And so when we want to move more non-invasive, like less use of X-rays or less bulky, like not MRIs, but bedside devices, which is the emerging trend. And we are very well in that direction. That's what I would say. Is we have a very good computational power, which is actually helping these non-invasive light-based technologies to be on bedside, not as bulky as MRI, nor as invasive as X-rays. So there is a huge revolution that is happening around science. I wouldn't say a couple of years, maybe decades it might take, but we are very well in the track. And computational power or the processing power is playing a major role here. Just to add, learning and artificial intelligence, that's playing another role because sometimes the variables are too many. It's too hard for us to be decoupled using physics. So we know physics propagation, at times it's not possible. So we work with certain companies where we use machine learning because it can handle multiple variables, though it doesn't have a knowledge of it. Of course, it becomes complicated. We need a huge data sets to test it. But if we mix both machine learning along with physics, known physics, it becomes much more less intensive, and we can achieve better results with small data sets as well. So yeah, that's my word on it. That we are all these things are converging, which is a very good
Converging.
news.
It's converging technology. That's true. So before I add anything, Dr. Saisham Narayana.
Yeah, I thought this is not my area per se, but see, I have seen in India there are companies right now, startups, in fact, developing technologies where I think image processing has become so important. Like I can give you example of companies actually having this whole slide scanners and like for detecting cervical cancer or oral cancers, and clubbing this with machine learning and artificial intelligence. So it's becoming a standalone diagnostics where I think can actually take away the subjectivity that a pathologist would. I mean, it can alleviate that kind of a subjectivity. So this kind of process, of course, I just want to bring into, I don't know whether it's relevant over here. I'm sure we don't have the optics, the bandwidth that do we have? I'm not very sure. Like when when it comes to kind of developing say endoscope, an indigenous endoscope. So there have been attempts in India. So from that part, I know that we really don't have the technology with us, and still we are importing some of those optics and CMOS and things like that. So I would like to hear from experts if any of you have any idea on, I mean, since it's connected with images and imaging processing, I was thinking if somebody could discuss that part also would be interesting to this audience. Thank you.
Yes. I have some inputs, but before that I will just go around the table. Ma'am, Vandana ma'am, have anything to say? Or Satish?
So, I mean, I just came across one paper. It was it was in 2018. It was regarding the endoscope. The endoscope is something like very, very thick, and taking it inside was is very difficult. So recently, what they developed is it's like a single pixel optical fiber, and they will be attaching the camera will be right at the end. So it's like a fiber. So the fibers and there will be light source, and then it is like you can have a scanning inside. So it won't give the image right at one point, but at one stand. So it has to be like a scanning inside. So you can move the fiber with your electromagnetic. You can connect the electromagnets, which can by controlling the current on it, it can actually move inside, and you can image the entire stomach. So that is what I have actually came across this paper. So it is published somewhere in 2018, and they were developing it. And definitely, this was not the Indian group. I think those were the European groups, but that is doable.
It is doable. Yeah. I mean, you're right, ma'am. This is doable. How I would not have told this last year this time. I always had this assumption that with respect to lasers and optics, India has not evolved in R&D. I would say some of them are true because we are still young in that culture. But I do have some few startups who can do their own OCT, and I do have a surgeon who has all the knowledge requires what endoscopy you do. And as I said at the beginning, doctors have the spectrum of clinical problems, and when they go and just bump into engineers who has solutions, they bring innovations. And this surgeon, he is a ENT, head and neck, and this surgeon, he came in contact with an engineer, and they gelled really well, and they're coming up with a robot-based endoscopic machine. And yeah, they got all the principles ready. So it's proof of concept is not, but this is just one example, right, of me sampling someone just in Visakhapatnam in Andhra Pradesh. I'm sure if I go around, I would search all day. All we need is an ecosystem, for them to meet. But I do see that. And another thing, answering to Dr. Saisham Narayana, there are a few companies who does this. They integrate equipment, say for an OCT. So there are 4 different items that go and make an OCT, an FPGA and spectroscopy and delay optics and some free space optics. All these make a OCT. So I know there are companies who are constantly depending on their vendors, and but in their background they know what technology goes in. Now they got so fed up, they started making their own now. So there are avenues. This is a possibility, really. So how much of these forums come in? It help. Yeah, please. Yeah.
Yeah. Thanks for that update. That's really nice to hear.
If I can add to your question, Sham, means so the endoscopes means I would say there are a couple of 2 things that we have to consider here. So one thing is the ingredients, of course, like CMOS detectors or CMOS cameras or certain niche area optics and so on, we may have to import. This is not possible because we don't have an ecosystem which can provide that. But what's even more important in healthcare technologies, the recipe. So these are ingredients, like the CMOS cameras or the optics and so on, they are the ingredients. But what is really making difference is the innovation. So when you look at an image, how do you know this is cancer or not? And how do you know this kind, if you probe it this way with this much light at this particular wavelength, with this much sampling rate, this will determine its cancer or not. So this is what the innovation is happening in endoscopes. It doesn't happen simply. You have to invest billions of dollars in research, where you can use, even if it is off-the-shelf technologies like CMOS, put them together and try to look for the niche area or try to innovate new applications. So the current trend is more modalities. It's not one. It's not just imaging with white light. It's multimodal. You put white light with OCT, with fluorescence and so on. So couple of technologies together and try to address a certain problem for a certain population, for a certain disease. So said this, we are not limited. That's what I want to say. Means we are not limited by anything. If we can innovate, even if you take devices from outside, in a long run, we will be holding the technology because patents will be staying in India, and we will be exporting these products. Like we will buy cameras for 10 euros, probably we will be exporting for 10,000 euros because you are adding your ingredient of innovation to it. This is where universities like Sathya Sai Institute of Higher Learning or big universities play a major role, where they can obtain funding on these niche areas from external industries or from the governments to create certain these kind of devices that can be, the mark of our country as a whole. So I would say we are not prevented by anything. We should be innovating. Every researcher we are sitting here, we should think big and try to see how we can get this happen. Yeah.
Thank you. Thank you very much. And since I'm more of an industrial background, I always have commercial mind, how things can be a product, and more than the, so far, after associated with the AMT, the thing is became more like an NGO all of a sudden. But just from people who have strong academic background are still associated with the universities, where do you think the funding or automation, funding opportunities is going on, and our students, say from not just Sathya Sai Institute of Higher Learning, anywhere, where are the scope? I mean, within this discussion, I picked up 2 or 3 topics, but that's just my opinion. I can tell you at the end what are they, I would say. But as kind of panel members, I would like to hear from you where the funding opportunities are.
Narayan, well.
Go ahead.
Dr. Narayan. No, I just wanted to hear from you because I want to know that where the fundings are.
Right. So can I take this question, and with your permission?
Please. Sorry, I should have. Yeah, please. Yeah.
Can I share my screen with a small presentation on from Bioactive? Thank you.
Just 1 second before.
Yeah.
Not issue.
Is that okay? Can I go ahead?
Yes. You can.
Thank you. So I'll be very quick actually, just to answer. Just give me a second. Yes. So is my screen visible now? No. Not yet. I'm sorry. Yeah. Is it visible now, my screen?
No, not yet.
Not yet? Okay.
Yes.
So anyway, I think I'll go without this. I think it's. I don't want to take any more time.
No, it's visible.
Is it visible now?
Screen is visible.
Yes. Okay. Its screen is visible. That's good. So I'll just present this. Sorry for that. Okay. So I'm representing BioAct now. That's Biotechnology Industry Research Assistance Council. So which is based out of Delhi. And it's part of Department of Biotechnology. So the question that we are discussing right now is where should you look for funding your innovations? So I thought I would just give a brief presentation of BIRAC, which, as I mentioned, and incidentally happened to be the foundation day of BIRAC. So it's been 9 years since BIRAC came into existence. So it was actually set up on 20th March 2012. So what BIRAC does is it supports now startups, entrepreneurs, academia, and SMEs to develop innovative, affordable biotech products, technologies to address the unmet needs. So when I say biotech products, definitely the medtech and healthcare already forms part of it. And so BIRAC actually works as a central agency or an interfacing agency connecting different stakeholders to actually create this ecosystem for medtech. So I'm only focusing on the medtech over here for creating the medtech ecosystem in the country. So without taking much time, I'll just take you to some of the schemes that we have within BIRAC where you can look for funding. So as students, I would further limit it down to only just for your focus. I mean, you may just focus on the first 3-4 ladders, steps in this ladder. So these are all different schemes which actually supports product developments right from ideation stage all the way up to market. So the first one, if you see here, it's we have a scheme called Sitare. So the Sitare scheme is mainly for students, those who have, so basically students who are at their PhD level, and those who have ideas to actually translate into potential products. So it's handled by an agency called Srishti Ahmedabad with along with BIRAC. They select students for this award and gives 15 lakhs and gives mentoring. Again, you have EUA platform. It's another kind of a fellowship for undergraduates as well as for postgraduates, including doctoral and postdoctoral students. Here again, it's a grant where you can look for developing your ideas. You have a fellowship per month, fellowship of 30,000 for undergrads and 50,000 for postgrads. So for undergrads, for example, you can form a team of 5 and apply for this, where you want to take your product into a prototype stage and things like that. Then we have SIP, that is under the BIRAC's first scheme. SIP is basically this is Societal Innovation Immersion Program, where we support SIP fellows, where a student can actually get immersed with a problem for a set period of 6 months or 8 months and identify the problems and identify solutions to actually mitigate that, technology solutions with support from clinicians and academicians. Then going ahead, it's bigger ones like BIG. It's a flagship scheme of BIRAC, which supports startups for developing proof of concept. And then we have AIR, that is Academia Innovation Research. So AIR, I think it has been very popular these days with academias, and it's kind of a it's again a scheme that supports early development phase. So these are all after you finish your research and you start translating your technology or start translating your research or IP into a market ready technology. So you'll be actually following a set technology readiness levels that you need to actually achieve at the end of each of these things. So academic industrial research, and then going ahead, you have a CRS scheme. It's a contract research scheme where you can actually bring in a industry who would want to take forward your product. So I will stop there without taking much time. So I thought I kind of...
While you're in slide, I have a question. Sorry about that.
Okay, please.
So what is a scope? I mean, 2 questions basically. What is the scope of this? Could anyone within biotech with a proposal they can apply, or what is the scope of this grant? What is not included? What subjects are not included?
So yeah, so I think if you go to the scheme guidelines, it can be more clear. So we don't exclude any. So it's all interdisciplinary. So we're not going to exclude maybe physics, because I think if you go through what kind of projects we have funded, it includes mostly we have funded medtech very heavily, especially critical components within medtech devices and contactless devices, software, medical softwares. So everything gets covered under this one. So I don't think that should stop you from applying.
So as long as there is some as long as there is some application in medical technology or biotech, this is we can apply.
Right. You're right.
Okay. No. And
I have this.
Yeah. Please.
No, I have a. Looking at this presentation, I just got one more curious question to Shyam. So, typically the technology evolution is more like the core or fundamentals of it will be built in university.
Yeah.
But if it has to reach to market, it has to be co-developed with a startup or a company which can take the burden and invest the rest of the money to take it to the market. So that means we need always a system where it funds not only university alone or industry alone, but together. So there is a consortium of universities and industries. So I don't know, do you have such funding here, or is it only for startups or university alone in your?
No, sir, I think you've answered yourself in this. So we fund universities basically to develop these products. So that the technology doesn't stay with the university, it comes out to market either through a industry or maybe a startup. Definitely you would need that at some stage of your product development. Academia is not going to actually take it all the way till product commercialization. So at some stage you need to find a right partner, and then from there on, I think we can also help you in technology transfer and filing IPs and things like that. But from there on, I think it's the industry that needs to take it forward and then scale up and reach the market.
My question is more like, what happens typically if we just give the money to universities? They never put effort to look for a company who can take it. So I was wondering, it's better to have them within the consortium, a company as well, who is also co-funded by whatever, maybe partially.
Right. Yes, sir. So in all of these things, so all the schemes that I've explained, like from Sitare, EUA, BIG. So you have business mentors. Actually, you will not be left alone to work in a university in a typical academic environment. You will be asked to. You will be given business mentoring. You will be connected with investors. You will be connected with other startups. So all these activities, I think I've listed some of them on the left side of this same slide. So I think this is part of our activities that Bioactive takes up. So it's part of building this ecosystem.
One question.
Sorry.
So is it? After it is granted, then it is like Bioactive takes over and then helps the university or the person who has applied for it. Helps to really mentor it, or they ask before that do you have mentor or not? So is this one of the criteria to get selected in?
Correct. So Bioactive, actually what Bioactive does here is when you submit a proposal, we look for whether you have a clinical mentor. If you're working on a clinical problem, you have a clinical mentor or not to actually take it forward. So that definitely happens. But once you're into Bioactive fold, we definitely help you with our set of mentors who would actually guide you through wherever there is a problem. So the mentoring happens along with the monitoring of the project. So we kind of guide you and connect you with the right people. Yes, we do that as well.
Maybe if there is a time, I just want to add something more. Fortunate or unfortunate, I happen to be a researcher for a very long time, but I'm also an entrepreneur and also work with multiple European projects as well as state level projects. So one of the trend I typically see is when you get mentors. So whenever we have only academic project, even if we have a kind of mentor panel or mentor board who are interested in our technology, most of the time it doesn't manifest because neither do they have the time nor willing to put the effort to take the technology to their company because they are lost in their own world. So the way it gets around in certain funding that I see, and it is very much workable, is where the consortium not only has university but also has the company already on board. Maybe the university is funded 100%, but the company is funded only 70% by the state agency. Since they are building the technology together, since company has already invested 30%, they end up taking the technology and trying to exploit it as much as possible to make it as a product and benefit from it. So I don't know, means of course I'm not here to judge. I'm just giving my whatever the small little experience that I saw. Maybe it's more biased in my way of looking at it. I just felt that was successful. Means successful even as well as in our company. When we are taking projects, means we take the strategic ones and we work with the university, but we ensure that we get enough sales through the product that we manufacture, means that we work together to retrieve all the investments that we made, and it becomes a lifetime product henceforth.
Yeah, I think we can discuss it, but it's a good suggestion. I think some of it has already been taken into. So this scheme takes care of it. So all that you have explained now. But I think we can discuss it. I think back to you, Dr. Anand.
Thank you, Dr. Sai Sham Narayan. This is, I think this is the climax was really good, though, especially that particular ladder chart. I mean, I do hear those grants before, but I never understood in that sequence. And especially you kind of gave a spectrum how ideation to POC, one set of grant looks after, and field testing looks after by another set of grant, and also manufacturing commercialization taken by. That was very good. I think if permits, please share it with the group of us so that we can probably I think the university should know this, if at all. Right. And I do have questions, but I really want to give time for the audience. Dr. Gauri Shankar, can we take questions from audience, please?
Maybe a couple of questions because we are short of time also.
Yes, sir. Yes.
Anybody from the audience?
Saram, sir.
Yeah, go ahead.
Sarah, please.
So, like maybe 5-10 years ago, there was this organization called Theranos. They came up something like an all-in-one device to diagnose it. Is there any chance of that type of device possible, sir?
Sorry, what device is it, Akhilji?
It's a device which can test for all the diseases. One single device which can test all the diseases.
I mean, when you say disease, I guess you mean they look at your vitals, like SpO2, heart rate, pulse rate, and then based on few data points, it will tell your health. It gives a health assessment score and tell your say if there is a HSA based on the health assessment. Yeah.
Actually, they use the blood sample. It's not successful. It's a fake product. Actually, it didn't success.
So it is the product you're talking about. It is invasive, isn't it? It takes blood sample.
Yes. Yes.
Yes, I mean the technology is moving where there are multi-para monitors, where even without non-invasively, you can based on number crunching of huge number of mathematical data, it can tell your what's happening in your body. For example, if you see your inhale breath, if you live in a very cold country, and you're inhaling the ambient temperature, which is like below 10 degrees, but if you always check your exhale breath, it's always like certain it's a room temperature. I think it's always hot when you look at your exhale temperature, but that exhale temperature, that exhale breath is basically telling something about your body. So there are sensors which, if you have a smart mask, which can tell what's happening in your body. So I'm just giving you one sample. There's a multi-parameter monitor based on just breath, breath, and there are some earlobe glucometers based on how the temperature of the ear. So this is possible, Akhil. That's what I could say. And very soon you'll see a lot of these products.
Just to add to you, Anand, to the question, means of course the Theranos was a big scam, and means your question more like you were asking whether that is possible. The technology only with future evolution will say whether it's possible or not. Means everything is possible provided you have answered the questions that are needed to enable what you are looking for. So they promise to monitor too many vitals with this very small sample of blood, and they can say too many things. but probably it wasn't possible then. But if it is possible now, we don't know. Means we have to look at what is the current trend in the technology. But we should always remember, whenever there is a startup, out of 100, only 6 or 10 succeeds, which means we are all trying to fight hard to prove that what we say is going to happen. And they went to extreme because they concealed all the data. But what is the right approach is you say what you have and give your pathway and your business plan, and saying like there is risk. That's how the investor comes, right? It's a high risk but high gain. So there is nothing wrong in failing, but we should make sure that we are open and clearly state our risk and project what it will look like when it is successful.
So I would like to add one more point to Dr. Sanatana's thing. So with the blood sample, actually you can measure a lot of things. So because the chemical, basically anything which you measure it has some chemical information. Blood is nothing but a chemical, right? So whatever you are eating, whatever you are breathing in, whatever. So everything is, the information is stored in your blood. And if you take it out and if you do the mass spectrometry, and mass spectrometry is one of the strongest tool to really do the chemical fingerprinting. And that will give you almost every information about it. But the problem is mass spectrometry is very, very costly. There is no one who makes mass spectrometer in India. Most of us basically the Indians who are working in this area, they try to import it, and that is, again, it is very, very costly. And there is not one simple mass spectrometer which can give you this information. It's like a combination of mass spectrometer which can give you the entire information about the chemical composition of your blood. And once you have this information, or the doctors have this information, they can tell you everything about you, basically. If they have a standard ready and if the reference ready, with that reference, we can actually figure out that what all diseases or what all things you have. Basically, a patient has.
Brilliant.
I have maybe one last question. Probably Mr. Satyadeep can answer this. Companies like Big Tech, and there's so many other companies which are working on this kind of medical products. Of course, has already Satyadeep has mentioned, there are a lot of challenges. They're looking at power conservation. They're looking at form factor and so many things. But if I'm correct, we are still relying on some of the ingredients to be still getting imported from foreign. But one side we are still we are talking about Atmanirbhar, we're making in India concepts. So my question is, how do industries like Big Tech and others, how are they looking at this scenario? And okay, newer and newer products are being developed, but do you think it is really going to be, it is going to become affordable for a common man, or is the cost going to go up only?
Well, it's difficult to say. Maybe in the future it will become affordable. So we still import a lot of components, like mainly semiconductors, and in India we don't have such fabrication houses. And for optical components, for example, we actually try to get it within the country. We couldn't meet the exact specs sometimes, so we are still relying on imported components.
So do you think that not exactly you, means not Satyadeep alone, but in general, so when the government is putting so much effort in this direction, should it not also put effort in equivalently in the direction where it can support the companies which are trying to make these devices, so that the import can also be reduced? So which means we should be able to set up a good optics related company, or get the technology down to India to make good lenses or mirrors or whatever we want, and things like that. So then probably the cost can go down, and also the speed of these devices coming out may also increase.
Yes, so the ecosystem is very important here for realizing the product and sustaining in the market. So, very often simple things, for example, even PCBs, you're not able to get good numbers in time when the demand is higher. So there needs to be support. So there needs to be a committed ecosystem which works together as a community with commitment towards healthcare.
That's true. Saisham sir, you have something to say?
Can I just add a point? So since you have talked about import substitution, so currently Bioactive has a call open, which actually invites proposals on import substitutes. So it's a call which is open till 31st of this month. So any of your technology which can fit into that, I think maybe you should visit Bioactive website and look whether it fits in over here. So we are only focusing on import substitutes in all different areas, whether it's medical devices, drugs, or pharmaceuticals, and all these different enzymes, etc. So I thought I'll just put it across.
Very good. I mean, that actually answers the second part of Dr. Gauri Shankar, his question, whether government is supporting. Yes, they are supporting. And again, I'm adding to what Sri Satyadeep ji mentioned. You're right. We need an ecosystem, and there are even companies are open. If you provide them a mandate, a business case. Why if in AMTC campus we have a company called Amphenol and Honeywell. They are one of the giants in sensors and PCB manufacturing. So both going to happen in India, in that too in this campus. And we have companies who who are also looking into having our own optics here. So we are so dependent on companies like Thorlax. Right, so we're importing so much on Thalwax, and we do have a company in Kerala, but still they're trying to match Hallmark. Yeah, everyone knows that. So yeah. So I think we're all aware. Even somehow, I think Dr. Saisham is a very good example. He immediately gave up with an answer. That there is import substitute is a scheme. So I think we're all starting to work together now. So yeah, I see hope in this. Yeah. So.
Yeah, thank you so much. Just,
any more questions? No, sorry.
One question which is there in the chat box by Shubhakanta. It says, if we are surrounded by too many technology and machines that we, then what about the psychological and mental effect on the human? I think he's mentioning in terms of, we were talking about a lot of monitoring devices. So I think probably has asked that question.
It's a very good question. So, as anyone has answer, I can share, but I don't want to be talking here. Anyone?
So I will add, because the same thing. I mean, when I keep checking my mother's vitals, so she says that being ignorant is always a bliss. So better I don't want to be checked. So whenever I check. So when her sugar level is high, so she says, please don't do that, because I'll not be able to drink tea. And I want to enjoy my tea. Yeah. So I mean, sometimes I feel that it is not also a good idea to really keep monitoring. Yeah. But yeah, so because we care about our loved ones, we need to do that.
Yeah. I do agree. There is a need where children's going around, and there were until last year we had the ability to stop them from being on screen for a long time, and now everything go virtual. So the situation themselves allow them to own a gadget, own a mobile phone, own a laptop. So a device like this, monitoring device like this would be welcome. But again, there is a fine balance. How much data we gather, how much we consist with the data results we get.
Yeah, just adding on to, I was just wondering, so we have technology solutions addressing mental health as well, whether it comes to depression. So it's again you're again bringing up a problem for which again there is such technology solutions. So I mean, it's so yeah.
Okay. Thank you, sir. Thank you so much. Yeah.
Wonderful. Thank you. Thank you very much, all the panel members, for taking this time And there is, in short, I can go into detail of everything we said, but I will, there will be, I think it's quite obvious that healthcare, there is definitely a lot of, leverage towards the healthcare, the technology. I see when I did my engineering, I always thought all these disciplines are separate. Now I see, like in this panel itself, you will see, amount of computer programmers, communication engineers, analog design engineers, biochemistry, molecular biology, laser optics, and and still we are not happy with it. We still wanted more. We want to go towards data analytics, and and blockchain is another, blockchain in medtech. That's also coming. Fintech, blockchain. That's another thing that's coming. So I think as a whole, I would feel we are all marching in the good direction, and I feel basically the green of this is what Dr. Saisham he shared. So the data you share, that there's a lot of support from the government. If we have the right proposal and right business case, and there's always a hope. There's always all encouragement from the government. Thank you very much, sir. Thank you very much. And so, Dr. Gauri Shankar, unless you have anything else for us to ask or share, we will close the panel.
Yes, sir. We are very thankful to all the panel members. It was really a very lively discussion, and I'm sure the students would have learned a lot from this entire exercise. And our endeavor was precisely that. And we hope that this would have really enlightened them on the various aspects and how the technology is being driven, and it is in what direction it's moving. So I once again thank all of you, sir, and special thanks to Dr. Anand for steering the discussions, and thanks to all the panel members. Thank you.
Thank you.
Sairam.
Thank you. Thank you so much. Sairam, everyone. Thank you.
Sairam. So to continue the session, morning we had 2 of our department presentations. So the remaining 2 presentations will also go on now for a short period of time, after which we will wind up the seminars, the 2-day webinar with a kind of not a validity session as such, but an overview of what has happened in the last 2 days, and then we will end the 2-day webinar. So I now request the third presenter from the department, Dr. Abhishek, to present on what kind of work he's doing related to biomedical, after which I request Dr. Muthu to present. So probably 10 minutes they will present, and after that we will have the final session. Thank you.
Sairam, sir. Am I audible, sir?
Yes.
Sairam.
Yeah, go ahead, please.
Yes, just sharing my screen. So 1 minute. Sairam to everybody. Offering myself at Bhagwan's lotus feet, respected guest speakers, respected teachers, and my dear brothers and sisters, a loving Sairam to one and all. Today I will just be giving a brief introduction to my work, which is on the computational and experimental studies on SERS, Surface Enhanced Raman Spectroscopy, for the detection of neurotransmitters. So just a brief introduction to Raman spectroscopy and SERS. As we all would know, there are 2 primary methods by which light interacts with the vibrational states of matter. One is Rayleigh scattering, another is Raman scattering. Of course, if it is absorbed, we would have IR spectra also. But in the scattering process, there are 2 different scattering processes in general. One is Rayleigh scattering, which is an elastic scattering, where the incident photon does not change in energy. And we have Stokes and anti-Stokes Raman scattering, which basically have a change in the energy of the photon. So basically, there is some exchange of energy happening with the molecule, and it is an inelastic scattering. And this is the Raman effect, which was discovered by Sir C.V. Raman in the early 1920s and 1930s. But what was found by him, even though it was initially realized that it was of great analytical potential, Raman scattering was not useful as much until the early 1970s, because Raman scattering happened to be very weak, of the order of 1 in 10 million photons would undergo Raman scattering. So in 1974, Fleischmann et al., their group discovered that the Raman signals intensity were multiplied manifold or enhanced manifold in the presence of a roughened metal. So this phenomenon came to be known as surface enhanced Raman spectroscopy, because the Raman signals were enhanced in the presence of a metal surface. And once this phenomenon was discovered, it was exploited as a sensitive and specific nanoscale phenomenon for detection. And you can see a graph on the right that typically demonstrates the comparison between Raman spectroscopy and ACRS. We can see that the Raman signals of 0.1 molar solution of rhodamin 6G. Rhodamin 6G is a standard compound which is used to study the efficacy of Raman substrates. So we can see that the signal is extremely low even for a 0.1 molar signal over here. Whereas on top we have the ACRS signal, which is a 1 micromolar signal, and you can see that the enhancement is very great as compared to the plain Raman spectroscopy. And this is the potential of ACRS. It can detect signals from compounds which can usually not be detected by conventional Raman spectroscopy at that concentration. There are so ever since it was discovered by Fleischmann's group, there have been a lot of ways of performing this ACRS experimentally. Historically, first they started with colloidal systems and electrochemically roughened silver. These were the first two systems historically. Of late, ever since nanoscience took over, metals nanoparticles which were deposited onto substrates have been gaining traction as one of the most popular methods of developing ACRS substrates. And here on the right, you can see this is taken from a publication which is a pen on paper approach. So the silver nanoparticle solution is concentrated and put as ink, and that ink is deposited onto an A4 paper. So this is a very simple technique of getting an ACRS substrate which can enhance the Raman signals. In my work, I had used a similar substrate where instead of using a pen, I just drop casted it using a pipette, and I could obtain a very easy way of obtaining an ACRS substrate. There are various applications of ACRS, right from detection of low abundance biomolecules, detection of proteins in bodily fluids, trace element detection, drug and explosives detection, and food quality analysis. The thing about ACRS is, as many of the esteemed speakers have mentioned, one of the desired quality of these point of care devices and for the upcoming technology is that it should be non-invasive. And ACRS is a completely non-invasive technology where you just need to put an amount of sample onto the substrate, and based on the efficacy of the substrate, it can show up signals for concentrations as low as what you can see here, 10 power minus 11 molar. And there have been cases, there have been applications where ACRS has been exploited for even single molecule detection. So coming to my work, I have worked on a biologically important a set of chemicals called neurotransmitters. So neurotransmitters are substances that are produced by neurons to transmit messages. Until the early 1900s, it was widely believed that neurons communicated with each other only through electronic messages. But the 20th century will reveal that neurons could communicate with each other even by chemical means. And these chemicals are released into what is known as the synaptic cleft. It was a small gap discovered between the neurons, and these neurotransmitters are released into the synaptic cleft and they bind onto specific receptors on the membrane of the next neuron. And depending on the amount of neurotransmitters that are deposited onto these receptors, so it can communicate what is the health of the person, particularly mental health, because the neurons are associated with the brain. So there are a variety of neurotransmitters. I have chosen 6 of these for my work. We can see there is dopamine, norepinephrine, epinephrine. Norepinephrine and epinephrine are more popularly known as noradrenaline and adrenaline. Dopamine is a compound which indicates the mental status of a person, and if levels of dopamine, norepinephrine, epinephrine are not proper, then they are indicative of diseases like Parkinson's and other mental illnesses. So I chose 6 neurotransmitters which are the most abundant in our human body, and these are computationally optimized structures of these neurotransmitters. So in my work, I have aimed to study the SERS of these compounds, both in a from a computational point of view as well as from an experimental point of view. So in the from the computational perspective, we calculated the Raman spectrum of these neurotransmitters as they are represented here, as well as in the vicinity of silver atom clusters. So I studied the Raman spectrum of these compounds, where silver atom, 1 atom, 2 atoms, 3 atoms, and 4 atoms were placed one after another, and we studied the Raman spectrum, how the intensity varied as we increased the the number of atoms in the silver cluster. And the result is shown here. So there are some specific modes which are linked to the benzene ring, the deformation of the benzene ring. So the vibration caused by that deformation leads to the Raman spectra, and we can see that as the number of atoms in the silver cluster increases from 1 to 2 to 3 to 4, there is a perceptible increase in the Raman signal for specific peaks. And this is indicative of the Raman enhancement in the presence of metal. So these were the computationally calculated spectra. I'm sorry, I'm not able to provide more details on this due to lack of time. We also compared it with the experimental spectra, and we can see that on the top, the orange color represents the SERS of dopamine at a concentration of 1 millimolar, and we are able to assign vibrational wave numbers to these and also compare them with the calculated wave numbers, and we could assign them to various vibrations. So that dopamine 1 millimolar SERS signal is compared with a dopamine 1 millimolar on fused silica. So for this particular spectrum, to obtain the SERS, I have used a fused silica based substrate where silver nanoparticles were deposited on the fused silica, and laser sintering was employed as a method to kind of stick these nanoparticles onto the fused silica. So in a very simple explanation, once the silver nanoparticle paste is uniformly pasted onto the fused silica, the heat created by the laser would cause the nanoparticles to aggregate and create small crevices within the fused silica. And this little bit, this amount of heat which is created causes the silver nanoparticles to get stuck onto the fused silica. Silica is nothing but like a small glass slide. So this is the spectrum which we obtained from that particular substrate. And that is compared with the same concentration of dopamine, but on fused silica without the silver nanoparticles. So this is conventional Raman spectroscopy. And you can see there is a stark difference in the shape of the spectrum as well as the peak heights. A lot of peaks are not seen without the phenomenon of ACRS. So we also went down in concentration and checked as to what was the lowest limit of dopamine we could detect, and we found that we could detect as low as 1 micromolar level of dopamine. So these neurotransmitters are important biological compounds, and once we perform such an analysis for other neurotransmitters and we can distinguish what are the peaks that are coming from these particular neurotransmitters, we could potentially employ them in a real life sample. If you have a plasma sample and you just put it on the ACRS substrate, based on the height of the peak, which is calibrated, we could detect the amount of neurotransmitters up to a certain level. So this is an application of a physics phenomenon, which is Raman spectroscopy, enhanced by a surface which is applied for a biologically important compound. I thank you all for your patience, and I would like to take any questions if there are any.
Yeah, I have one question.
Yes, sir.
Is 785 the most preferred? How about 1064 nanometers?
Right now, the Raman system which we have is a 785 nanometer laser. Particularly for biological compounds, 785 is very good because it is away from the fluorescence peak of bio compounds. Usually biological compounds have their fluorescence peak, that is their absorption peak, at somewhere around 200 or 300 nanometers. So 785 nanometers is good, sir. I don't think it having a 1064 nanometer would have a significant improvement over a 785 nanometer laser for such compounds.
Okay. Second thing, this is just a comment, like
Yes.
Like in a lab, you can basically use purified samples and get the Raman spectra.
Yes, sir.
But when you are extracting something from a patient and trying to read that using Raman,
yes, sir.
I think there will be some challenges like interference or contaminants which could corrupt the
Yes,
Raman.
so studies done on plasma and real-time imaging of ACRS. So basically, for a given substrate, the way you fabricate the substrate, there will be certain peaks of the compound which will shoot up. For example, in this particular ACRS, we see this 949, we see the 1385 and 440 and 1607. So I am just choosing some 4 peaks which are becoming extremely prominent. So once 3 or 4 such peaks are identified for one particular substrate, again, if I fabricate a different substrate, the shape of the spectrum could be different. So we need to study these with pure samples for sure. But once we study these and identify 4 or 5 different peaks that are going to be the signature peaks of the compound, we can use these for quantitative analysis once we do a calibration. this has been reported for other standard substrates earlier, sir.
Thank you.
Sir, shall I stop presenting?
Thank you, Abhishek, for making my introduction to Raman spectroscopy easier.
Saram, sir.
Saram. We'll continue with the next presentation. So I have all the advantage of being the last speaker, so I can synthesize all my talks with reference to all the previous speakers, but want of time and disadvantage to the students and the audience, I'll just have to go through some of this. And any questions, I could like you to post it to me. Offering myself at Bhagwan's lotus feet for giving us this beautiful ecosystem of having this educare and healthcare, having a wonderful symbiotic relation at Puttaparthi or at Bangalore, where we have these two hospitals and university labs working hand in hand and developing so many things that itself is a unique privilege to us. I also thank all the doctors, clinicians, the people who built this ecosystem before I venture into this talk. I like to make it more lively for the students, and so that's a pace I'm going to set in. So most of you are familiar with all the things that I put on this slide. We have Pediasure, we have Boost, we are but not sure whether our immunity will boost. And many times our food market comes into limelight. Especially I could also involve or take reference to Vandana madam, who is building a mass spec in the Maggi noodles, which is one of our favorite food for our researchers or in the kids, had lot of amount of lead. In all these scenarios, what comes into rescue is the analytical power of the technologies that we built. Whether it could be a drop of blood of microliters or parts per billion of lead present anywhere in unwanted cases, that could be all the demerit that I could tell somebody could take advantage of making more profit. But when we start looking at all these things, which we are at a crossroad where whether we wanted to have a healthy life or come out of an illness, so many times what comes to our rescue is the technology. Either way to take it. So today I will be dealing with something called bone necrosis, which was a wonderful work. A lot of people worked, and how to probe this bone necrosis is what the work that our department took, and it was part of one of the PhD students from bioscience. It was an interdisciplinary project, but the clinical relevance made it not only for the sample but also for the remediation that we need to intervene for the patient care came from SSIHM, our hospitals. So in the afternoon talk by Professor Oken, you heard a lot of things that we were doing outside our body. We could have the whole organ, any part of the organ nowadays getting built synthetically and substituted. Most of the time, this necessity comes when we are facing a traumatic situation. But the human body is a very wonderful and a complex machine. It has this magic to something called regenerate and heal. So one such thing that happens many times to us when we are kids, or many times we tend to have an opportunity to see is the self-healing power of this bone. So a lot of these bone or biomaterial was discussed in the afternoon. So I would like to take you something like this, a very easy way to understand. As some very good question from the audience came, can this material grow as we grow? The material that you substitute in the bone. Can it also resorb?
Lot of questions were there. So we can connect it to something like the road that we lay. This process of bone, how it grows, is like making a new road. Correct? We all are familiar with the tar and the metal that they mix and make this. Exactly the same thing happens in the bone surface. We have agents. We have the civil construction and person. And at times we need to wonder whenever we have this trauma or when we became age, or there could be sometimes something called as idiopathic fractures. We don't have any reasons for having a fracture at all. It happens. And many times we also go on to relay the road. So this is the magical part where you may not like to do an implant or substitute, but you want your body to heal by itself. So this sets the tone for the biologist, an exciting area. So how can I enhance this self-healing power? Otherwise, we land up in degeneration, which many often we see in our aged people, or when we also see in our first slide when we are talking about can I get a bone density test free? So this is where our scientific pursuit ventured. It's not. So we wanted to understand what is a bone and how these fractures are happening. So the morning or the afternoon talks were so much elaborate on the material that we are substituting as a bone. So I like to take you to an ultrastructure of a bone. There were some questions on whether nanomaterials could be used there. So first we need to understand what is the bone at a macro level, a micro level, and at a nano level. So if you see a bone, you can all see this. It looks like a trabecular meshwork that you see very often. It's more like in the Tom and Jerry cartoon that you see a cheese piece. It's a lot of things. Structures are there, a meshwork. As you see the building block of the bone, as you go further, take a clockwise thing, you land up something called as an hydroxyapatite nanocrystal. And then you have the collagen. These two things actually makes the bone and gives all the mechanical strength that you derive from a 36 kg bone. That professor explained that it was taking such amount of stress and strain. How does it happen? So the magic happens at this micro and nano domain, where in which you have the collagen, which is the tissue, soft tissue, and the mineral, which is going to give you the hard tissue. So coming to the bone as such, can I have next slide? So if you go on looking at it, this is a hierarchical structure wherein which you have this slowly when you come towards this, you have the proteins, you have the collagen, the marriage between the mineral, that is hydroxyapatite, and the collagen. So this is more like a sleeping mat, very closely knitted, that gives you the strength instead of a single thread when they are knitted together. So this is the magic that happens in our every bone cell. So now the question, what is the scientific question we were addressing? We were looking at a particular fracture called femoral head, that is nothing but your hip joint. So if you wanted to go back and analyze this complex structure, you need many things to prove. Why are we having some vitamins added to our Boost or Pediasure, or even in your cooking oil? Why are they adding vitamin D or E or B or a B complex? Where is the need coming from? So many times when our body grows naturally, or if you are deficient of these vitamins, you lack some of these amino acids to produce these minerals, to make this mineral or make this structure. So whenever we are going to have a lack of this structure, slowly what happens? The relaying of the road doesn't happen. So as you grow, or whenever you are going to a stress, you need a self-healing process to happen. So this is where the bottleneck is. So this is the domain of biologists, which I will not step in. And this is where they talk about the metabolism or the failure of a metabolism. So I leave it to my colleague who will do that. And to such a complex problem of looking at the bone tissue as such a hard tissue, we wanted to finally understand that this is the cause, a deficiency of B complex. Our goal was to find out how this is happening. So we wanted to see the cause, we want to see the effect. But how to measure a vitamin B complex? So those set of experiments were done by our biochemical data, which could be from your body fluids, urine, blood samples, which went through mass specs, what Vandana madam was referring earlier. Then on the other side, we have physically seeing is nothing but believing, correct? We need to really see at the site where there was a diseased bone and look at it, what happens at that. Those things came from the immuno and histopathology. And then we had the radiology people explaining to us without destruction and how do I look at the bone, we had the micro-CT coming in, tomography. But all said, then we need the bone samples. We cannot butcher a person to take a sample, correct? We need this bone sample. We want healthy things. How do we take this? So this is where our hospitals had given us those bone samples from the traumatic patients because they might have got a fracture from an accident. So those people, patients, in a way, whenever we work in biology, we should be very thankful to the society because though you are going to serve the society, we are going to take all the help from them, from their simple sample consent to everything. It's a very, very ethically, one has to tread very carefully to do that. And this is our Ashwat Narayan brother who is working on such sample and collecting a micro-CT. And what we did was this electron microscopy and micro-Raman studies here. So I will now move on to show you how this bone looks. So you saw the ultrastructure there when I showed you. So it's a cross-sectional view of a bone of your femoral head, your hip joint basically. So it's a cross-section. So one thing I should thank our doctors of the hospital because if they get a trauma patient anytime, because most of the time you'll end up in odd hours of the day, they could give us a call, we go collect the sample. We have to take the consent after the operation. We take this, keep it in our refrigeration, deep cold refrigeration, thaw it and immediately do. Because if you delay by any hour, all that biochemical information that you are going to probe is going to be thrown directly into the ditch. You won't get any useful information. So we wanted the sample as close as to the reality and see the biochemical things. So where does Raman come in? So Abhishek had shared about the Raman process as such. What I wanted to tell you is these are the two spectrometers that we have in our campus. So one of that is a dispersive, both are dispersive Raman spectrometer, but one is a spectrometer, but another one is a micro spectrometer. And I will also like to highlight that these facilities by several by blessings of Swami and several agencies which funded us, including our central trust, we could get all these. When we started the research on this bone, we had only this setup. It was very difficult for us to do because you wanted to go at a micro scale and look at what is the biochemical fingerprint or the molecular fingerprint. You can never do this. You have to couple it to a microscope. This was almost a $500 million facility that we got from DST FIST funding. So more or less, what I wanted to tell you is these are the two experimental setups that we used. Now it's a very interesting thing. So why does a bone get a fracture? Either it could be an accident, but many times when we see our aged parents or aged siblings, we know that many times they undergo a failure, hip failure or a spinal failure. They have been working, just that stress and strain in their job, or it could be just their age that changes the ultrastructure of the bone. So what we were trying to look at is, where does Raman help in? So what I am projecting here on the left panel is the Raman spectrum of an control, what I could call it as an healthy bone. So morning when, afternoon when the professor was talking, he was talking about an hydroxyapatite mineral. So the mineral hydroxyapatite is nothing but calcium phosphate. So calcium phosphate is a strong component of your bone matrix or the bone as such. So the bone is built up of mineral and matrix. The matrix is nothing but your collagen, which is our soft tissue like our skin, and the mineral. So the mineral. So if at all any damage that could happen for various reasons, then what we should be getting is a variation in these peaks. As a physicist or a spectroscopist, area under the curve could give you the intensity and which you can strongly correlate it to the concentration of the material that is present. That's all. And what I would like to tell you is, many times, most often, the solution or the metabolomic query that we had is why does the bone failure happen was related to amino acids which were not getting produced in your body. So that's where you take the vitamin B complex, which is a simple solution to the complex problem, wherein which a B complex could fill in all the metabolism and prevent a bone from becoming brittle. So what is it brittle? How do I know the sample that I am analyzing is going to be brittle? What I have is nothing but the phosphate peak will go down. Basically, the bone is not able to restructure itself or relay itself. So what happens, the phosphate content or the construction problem, we are not having the raw material for constructing the bone. What gets substituted is carbonate peak. So what do you have? Carbonate is nothing but what is popularly a calcium carbonate is nothing but your chalk. So your bone becomes a chalk piece. And all of us know how brittle it is. There ends the story. So we just had the advantage, rightly as somebody put it, to try this, what really is a necrotic bone different from a control bone. So we could run with it. As Satyadeep asked somebody the 785 nanometer, we could see all the fluorescent signal coming, getting the electronic transition with the Raman signal here. So that time we really did not have the Raman spectrometer with us. Swami's grace and devotees from Raman Research Institute shared their facility to venture into such a useful problem for us. So this is what. So we really put the bone under the microscope, the Raman microscope there. So now you can see a healthy bone, but it has a necrotic part here. The necrosis is nothing but, many of you have seen in the bone ultrastructure, there could be veins running into the bone. Because bones are the bone marrow is the birthplace of all the blood cells that you have in your body. So if the bones are not going to get supplied, the necrosis started. This is like not watering your plant. Exactly when there is no blood and the required vitamin B complex in your bed, then the necrosis starts here. So what we smartly started doing was doing a line profile on the bone sample. But by Swami's luck, we could also have our micro Raman installed in our facility. Then we could do a mapping itself. So we went on to, instead of a line scanning, we went on to do a mapping, chemical mapping. So what I could show you was, if at all the phosphate has disappeared from your bone, then I should get more carbonate peak. So it became a simple relation for us to pick up. We can directly view where the phosphate was located, phosphate bands were located. So what you're seeing is the heat map of phosphate in avian and necrotic area bone. Avian is nothing but the avascular necrosis, which is the scientific name for the necrosis that is happening in terms of biology. So we could get all the radiation, all the maps, heat maps on this. So this was the window of helping Raman spectroscopy to do that. So I would like to highlight very visually, very easy for you to perceive is that the heat maps given here. Wherever red is there, you could see a very high index of this mineral matrix ratio to be present. So that mineral matrix ratio is how much of percentage of phosphate to my collagen ratio. So if I have a large phosphate, already I know I am a healthy patient here. Okay, I am not doing on live samples, meaning on the live patient here, I have borrowed those control samples who do not have avascular necrosis. And on the contrast, you see the blue color, you see all these patients are having very less patient samples basically. In a very 2D image processing way, you can come to this.
And we could also go on to tell that if at all collagen itself is not being produced, the next problem is mineral is deficient. You could give for a very substitute it with calcium or phosphate that you take in your Horlicks or in your, most of the time in the women health drinks, we have this phosphate or the calcium substituted there. But how do I know that my collagen is not working? Then I have to look at the other ratios too. So there are some more peaks which I have not showed here probably. This is the carbonate phosphate ratio also here. So I will end my talk highlighting one thing, that whenever you take real life problem, it is never one department. It's always inter-department. It is inter-institutional. It is sometimes across the continents. And all that we have with us is a selfless passion and a help and a love for serving the society. I could end with two of my mentors who had actually helped us doing Raman spectroscopy and who never cared to put their name in this wonderful work, Professor Rao and Professor Arun Srikumar from Baylor College of Medicine. They put the whole team across many continents here, which I think by Swami's grace had a Nature Scientific Reports publication. I thank everyone for this opportunity.
Humble pranams at Bhagwan's Divine Lotus Feet. Respected elders, present physically and also virtually, dear colleagues and dear student brothers. Sai Ram to one and all. Thank you, brother Dr. Muthukumar, for giving us a very nice overview of the work which the Department of Physics had undertaken. Just last year, our Ashwath Narayan had finished this work, which brought a good credit to the Department of Biosciences and also Physics. Now, I was asked to give few concluding remarks. I'll make it really few. We have successfully completed this 2-day international webinar on physics for healthcare. I'm sure that all the faculty members and also the students have immensely gained from the rich experience of varied speakers, very, very eminent professors, industrialists, doctors. All of them have shared their rich experience, and it was really wonderful to be present and then learn these simple physics principles, how they are applied in the real life situations and specifically in the healthcare industry. I'm reminded about the vision which our revered founder chancellor had for all the students and the faculty of this great institution. He said, this university should produce students of good character, but with sound academic background. And that can happen only when we have a vibrant academic atmosphere. And that comes through a sound curriculum, seminars, webinars, and interaction with the industry personnel and also the healthcare personnel. Vidya and Vaidya, the education and the healthcare, are two very, very dear projects of Bhagwan. The Vidya project has taken the shape of university and also the number of schools run by the Central Trust and Vidya Vahini and so many Vidya Jyoti scheme and so on. The Vaidya aspect of Bhagwan is translated into the super specialty hospitals, mobile hospital, general hospitals, and all the medical care which is given by the Sai centers all over the world. So these are two very dear projects of Bhagwan, the revered founder chancellor. And then this particular webinar beautifully combines this Vidya and Vaidya in the form of healthcare, physics for the healthcare. No, that's a fantastic conception, and it all started with, stage setting by the HOD and the associate head for this webinar, and the message delivered by the Vice Chancellor, Dr. C.B. Sanjeevi, and also the chief guest, Dr. Relu Swaroop, the secretary to the Department of Biotechnology, was very clear. He said, there's so much of physics which you can apply in the healthcare industry. We just only need to open our eyes and innovate and find new technologies. We are happy that in spite of the pandemic which we are going through, our academic activity did not stop. In fact, thanks to, I would say, COVID, it has enhanced it, augmented it, because the digitalization has swept the country. Everyone now is utilizing or reaping the benefits of the digitalization technology, the digitalization which has happened in our country. This 2-day webinar, we've learned so many things, and definitely it has increased our amazement for the concepts in physics. Whenever we learn a new concept, we should now think of, see where it is applied and how it can be applied, or how it can be implemented in our, for our society. We have also seen the research work, the biomedical research work which is going on in our own department across the campuses in this particular area, because the department has identified this as to be thrust area a few years back, and we are slowly but steadily moving forward. And these 4 presentations, excellent presentations, were made this morning and this evening are a testimony for that. I have picked up a lot of jargon and a lot of important concepts in physics, and also new ideas from this webinar. I'll just only mention, this will summarize all the talks which were given by various speakers here, very eminent speakers. Things like affordable healthcare, non-invasive technologies, spectroscopy and its applications, computational work, radiological physics, accelerated physics, biocompatible ceramic materials, multi-photon spectroscopy, AI, artificial intelligence, ML, machine learning, deep learning, DL, Raman spectroscopy, photonics, diffuse optics, adaptive optics, neonatal care, CT, MRI, SPECT, PET, engineering physics, OCT, and so many of them we are exposed to. In fact, the basic principles of all these things and more are actually there in our curriculum. So whenever we come across these things, pay attention to grasp them, and always try to see where they can be applied. I hope that efforts made by the Department of Physics should not terminate at this point. Rather, it should only enhance our understanding, our zeal to learn new concepts in physics and applications of these concepts in our daily life. So at the end, I would like to say, let us celebrate. Celebrate not because the webinar is ending, nor because it is Sunday tomorrow, but because we are students of physics. We should be happy that we are students of physics, and even teachers. Students are students only for some time, but teachers are eternal students for their life of physics. So let us celebrate because we are students of physics, and physics has so many applications in the healthcare industry, and we have seen just a sample of that. So let us resolve at the end of this webinar to study physics with greater enthusiasm, with greater zeal. And whenever we learn a new concept, let us ask, let us think where it can be applied and where it can be implemented so that the society around us gets the benefit of our knowledge. So, in conclusion, I thank Bhagwan for setting this beautiful ecosystem, as Brother Muthu put it, for us to learn these beautiful concepts, how the physics can be applied to one domain. Only we've just taken only healthcare domain, but it can be applied for so many places. But in this itself, we have seen that so much can be done, and we thank Bhagwan for making this webinar a successful one, opening our eyes and inspiring all of us to learn physics with greater vigor. Thank you very much. Now I would invite Dr. Vijay Sai from our department, faculty member from our department, to formally propose the vote of thanks. Sairam.
ॐ सहना भवतु सहनौ भुनक्तु सह वीर्यं करवावहै तेजस्विनावधीतमस्तु मा विद्विषावहै ॐ शान्तः शान्तः शान्तिः. The inner meaning of this Shanti mantra is, let us grow together in harmony, let us move about in friendliness, let us spread together the light that we have gained from our studies. Let us live in harmony without discord. Let us promote in harmony the use of our talents and skills. This is the profound inner meaning of the Vedic hymn. Thank you so much, Swami, for guiding us throughout this two-day international webinar. The organization of this event is solely due to your immense blessings and your guidance at every moment. Swami, please accept our collective gratitude for making this program successful and beneficial to all. We sincerely believe that only with your benevolent love, grace and blessings, we can achieve anything either individually or collectively. It is said that the essence of all beautiful art is gratitude. The best and beautiful things in the world cannot be seen or even touched. They must be felt by our hearts. Thank you is one such prayer among them. Life's more persistent and urgent question is, what are we doing for others? Or what is our contribution to the society that which we are part of? As said by Martin Luther King. Maybe he must have probably got this thought from our Vedic and Puranic dictum, Paropakardam idam shariram. The theme of this webinar, Physics for Healthcare, rightly fits into Science for Society. This webinar featured a series of talks by top academicians, researchers, entrepreneurs, and innovators from across the globe on the application of physics to diagnostic and therapeutic aspects of healthcare. On behalf of the Department of Physics, Sri Satya Sai Institute of Higher Learning, I consider it as a great privilege to propose vote of thanks to all those who have directly and indirectly contributed to this two-day webinar on physics for healthcare. All of us really felt that it was really a memorable, successful and useful event. At the outset, we would like to express sincere thanks to our respected Chancellor Sri Chakravarti Garu and also the members of Central Trust for their encouragement in conducting this webinar. I thank our revered Vice Chancellor Sir, Professor Sri B. Sanjeevi, who had very actively and enthusiastically motivated us and has given valuable inputs for this webinar. Sir, a special mention about your introductory remarks, which set the right momentum for the webinar. Thank you so much, sir. I also would like to mention a deep sense of gratitude to Professor B. Sai Girdhar, Registrar, Sri Sanjay Sahani, Controller of Examinations, and Dr. G. Raghavendra Raju, Campus Director, for their kind support and encouragement. Thank you, Dr. Renu Swarup, Secretary, Department of Biotechnology, Ministry of Science and Technology, Government of India, for readily consenting to be the chief guest for this webinar and for your precious time in delivering a very pertinent inaugural address. Our sincere thanks to all the distinguished speakers and resource persons from India and abroad who had readily accepted our invitation and given very interesting and thought-provoking talks. I would like to name them. I hope I will not leave anyone. To start with, we had Professor Dr. Khan Kiyu from the University of Arizona, US. Professor Dr. Fanindra Yelavarthi, one of our illustrious alumni from Indian Institute of Science, Bangalore. Dr. C. Murali Krishna from Tata Memorial Center, Mumbai. Professor Dr. Antonio Pifferi from Politecnico di Milano, Italy. Dr. Vyas, again, one of our illustrious alumni from the Stanford University School of Medicine, US. Shri Shashi Kumar, MD, Phoenix Medical Systems. Dr. Shuvan Prashant Turaga from Atomic Systems Private Limited, Singapore. Dr. Anshuman Jyotidas, Labby Inc. from US. These two were also alumni from our SSIHL. Dr. Shashwati Singh from Baba Atomic Research Center, Mumbai. Professor Dr. Hakan Enquist from Uppsala University, Sweden. Dr. Chaitanya from University of Maryland School of Business, Medicine, US. And to the panel discussion members, to the distinguished panel discussion members, Dr. Anand Krishnan from AMTZ, Dr. Sanatana from Tyndall National Institute, Ireland, Dr. Shyam Narayan from DBT BIRAC, Dr. Vandana Sharma, faculty from IIT Hyderabad, and Sri Satyadeep from Big Tech Bangalore, and also thanks to our department presentations by Sri Dani, Sri Abhishek, Dr. Murali Ravi, and Dr. Muthukumar. All the talks were really good and were pitched at a level which our students could understand and appreciate. Thank you all the panel discussion members for sparing your valuable time and sharing your knowledge on the very relevant topic, the changing landscape of healthcare technology. Special thanks to Mr. Raghuram for all the support at this Multimedia Center. To all the faculty and research scholars who had worked tirelessly to make this webinar a successful and useful event, a big thank you. Thank you all the students from the three campuses, Prashant Nilayam, Brindavan and Anantapur, and all the alumni who had actively participated in the webinar through YouTube, Facebook, and other social media portals. Once again, I thank you all, Sairam. Let us close the webinar event with Mangalarti to Swami. Sairam.
Om Jai Jagadish Hare Swami Satya Sai Hare Bhakta Jana Samrakshaka Bhakta Jana Samrakshaka Parti Maheshwara Om Jai Jagadish Hare Shashivadana Shrikara Sarva Pranapate Swami Sarva Pranapate Ashrita Kalpalatika Ashrita Kalpalatika Apad Bandhava Om Jai Jagadish Hare Mata Pita Guru Daivamu Mariyantayu Nive Swami Mariyantayu Nive Nada Brahma Jagannatha Nada Brahma Jagannatha Nagendra Shayana ॐ जय जगदीश हरे। ओंकार रूप ओजस्वी वो साई महादेवा, सत्य साई महादेवा, मंगल आरती अंदुको, मंगल आरती अंदुको, मंदर गिरिधारी, ओम जय जगदीश हरे। नारायण नारायण ओम, सत्यनारायण नारायण नारायण ओम, नारायण नारायण ओम, सत्यनारायण नारायण ओम, सत्यनारायण नारायण ओम। ओम जय सद्गुरुदेवा। नारायण नारायण ओम, सत्यनारायण नारायण नारायण ओम, नारायण नारायण ओम, सत्यनारायण नारायण ओम, सत्यनारायण नारायण ओम। ओम जय सद्गुरुदेवा। नारायण नारायण ओम, सत्यनारायण नारायण नारायण ओम, नारायण नारायण ओम, सत्यनारायण नारायण ओम, सत्यनारायण नारायण ओम। ओम जय सद्गुरुदेवा। ओम शांति शांति शांतिः। समस्तलोकाः सुखिनो भवन्तु, समस्तलोकाः सुखिनो भवन्तु, समस्तलोकाः सुखिनो भवन्तु। ओम शांति शांति शांतिः। जय बोलो भगवान श्री सत्य साई बाबा जी की जय।